in the right direction.
Operant Pharmacy Federation is a hub for networking, support, engagement, and knowledge transfer.
Actually, the target was there. Pharmaceutical bits were added to my campus. I came to know about Operant Pharmacy Federation when I was in bachelor's degree. So when I did an association with them, I actually invited me and amazing fact to interact with pharmacy researchers and the leaders from all across India. I was that helped us achieve our ideas and gain knowledge.
And now, OPL has signed 20 plus MOUs, organizing more than 20 national and international conferences in India and abroad, including PHARMANEST and PHARMANEST, our two major annual conferences. We have also organized more than 75 workshops, symposiums, webinars, and skill development programs with more than 200 experts from the industry and academia. We are the first non-profit organization to issue e-certificates on digital order with over 30,000 delegates in attendance. As the country enters a new era, we hope to become a vibrant symbol of humanity's triumph. A smile is powerful and universal. Smile is infectious. It spreads joy and hope. And behind every smile is help. Our commitment to ensure health to Operant Pharmacy Federation. Powered by residents for emerging innovations. The Operant Pharmacy Federation, headquartered in Pali, Rajasthan, India.
The pharmacy reason to provide necessary skill set to the young professionals in the area of research, development, and innovation. Because there is a large gap between Indian academics and industry. We at OPF are committed to closing it.
We at OPF are a progressive, resourceful, committed to quality, sustainability, and transparent organization. Innovation is more about creating revolutionary new products and services as compared to evolution, which focuses on improving an existing item. We believe that often an innovation has the potential to disrupt our market by changing status.
In the past 4 years, OPL has organized many more skill development programs with highly skilled experts guiding the profession in the right direction.
Operant Pharmacy Federation is a hub for networking, support, engagement, and knowledge transfer.
Actually, the target was here. Pharmaceutical bits were added to my campus. I came to know about Operant Pharmacy Federation when I was in India.
Very good morning, one and all. I welcome you to day 2 of this conference on artificial intelligence, drug development, and drug discovery. Today, we are starting with plenary session 3. And to do the proceedings, I would like to call on stage the 2 chairpersons of this session. At the outset, I invite on stage the professor and principal of BGS College of Pharmacy, Dr. Narendra, onto the dais. Sir is also the secretary of APTA AP State Branch. Sir has a degree in PhD from Pharmaceutical Analysis in the year 2013 from Andhra University. Completed Pharmacy in the year 2008 in Pharmacognosy and Phytochemistry at Andhra University. Sir received Best Principal Award in the year 2021. He has more than 50 publications, 2 books, and also had 7 patents in his name. He also had an AHAT grant of 2.4 lakhs for the conduction of a conference. Next, I would like to invite another chairperson, Dr. T. Vinay Kumar, onto the dais. Sir is currently working as Principal, Victoria College of Pharmacy. Sir completed his PhD in the year 2014 from Andhra University in Pharmacology. He completed his Masters of Pharmacy in the year 2009 from SRM University, again in Pharmacology. He is having a total of 13 Indian patents and 3 international patents. With this introduction of the chairperson, I request the chairperson to please introduce the speaker.
Morning, Monitor. I take the privilege of inviting Professor Dharmendra Ahuja. He is a highly esteemed individual who holds a variety of roles in the healthcare and engineering sector, demonstrating a strong commitment to the both fields in India. Here is a breakdown of the key positions and recognitions mentioned in my speech. He is acting as Principal Mentor, Bureau for Health and Education Status Upliftment, India. Professor Ahuja is serving as a Principal Mentor for this organization, which is likely dedicated to improving the health and education outcomes in India, possibly focusing on marginalized communities or regions. He is a pattern member, Indian Pharmaceutical Association. Title suggests that Professor Ahuja is recognized member of the prominent national organization for professionals in the pharmaceutical field, likely contributing to policy, research, and development efforts in pharmacy. He is a member, Board of Management and Directors, Pharmaceutical Sciences, JV Women's University, Jaipur, Rajasthan. Professor Ahuja holds a leadership position at a university in Rajasthan overseeing pharmacy sciences. This role involves significant academic responsibility, possibly guiding curriculum, research, and academic policies. He is also serving as mentor for change, Niti Aayog, Government of India. This indicates that he is a mentor under the Mentor of Change program by Niti Aayog, a national think tank. This initiative likely involves advising and guiding young entrepreneurs and innovators in India. He is also an empaneled and assessor at National Assessment and Accreditation Council, that is NAC. Professor Ahuja is appointed as an official assessor for this council, which accredits higher educational institutions in India. His role likely involves evaluating educational institutions to ensure they meet required standards. Coming to few recognitions and awards, Professor Ahuja has received recognition and award from several important figures in Government of India. Health Minister, Government of Rajasthan, acknowledgement from the State Health Development Department for his contributions. Women's and Child Development Minister from the Government of Rajasthan, recognition for his work related to women and children development in the region. He is a Mayor, Jaipur, recognized local recognition for his contribution to community and possibly related to health education and social welfare initiatives. I welcome Professor Dharmendra Ahuja onto the dais for giving the lecture and detailed report on practical applications of AI tools. I welcome Dr. Ahuja onto the dais.
So a very good morning to all of you. First of all, I would like to thank PHITS management and of course Operant Pharmacy Federation for organizing such a beautiful conference. And it's a matter of pleasure for me to associate with this conference. I would like to thank the session chairs for giving brief introduction of mine and inviting me for the session. Of course, my love, Chandramoulipuram sir, who is always associated with me, and all dear students. So, we will be discussing today practical application of artificial intelligence tools, XPharm and XPhysio software. In your pharmacology study, you must be using also in your institution the XPharm software. And the principal mentor of the same is in front of you today. So, it's not a one-day thing. It has evolved from last 20 years, one by one, regularly modules added, and today it is the brand which is being recognized by more than 25 countries. More than 25 countries are using, we are having tie-ups with different governments also. And today we will be discussing practical application of AI because the conference theme is based on artificial intelligence. Because now the education is moving towards artificial intelligence. The classroom teaching is now moving towards AI teaching, software teaching, newer applications teaching. So this is the right platform. I would like to thank the CHIPS management that they have organized such a beautiful conference for you, which will really increase your skills in AI. Because in future, when you will be going in industry or anywhere else, your practical knowledge, your technical knowledge will be very much useful for you. So this is the overview. Like first we will see the introduction, then we will see what is virtual experimentation in pharmacology, what are the virtual experimentation techniques, what are the general software used in pharmacology and physiology. Then we will be seeing details about XPharm and XPhysio software, and then we will be seeing advantages of virtual pharmacology. So I got to know that yesterday also sessions were there. So before going in deep, we should know like what is AI, what is artificial intelligence. So artificial intelligence is a set of technology that enables computer to perform a variety of advanced functions, including the ability to see, understand, and translate spoken and written language, analyze data, and make recommendation and more. I mean to say, artificial intelligence is a next level of software programming. See, earlier in older days, we can say calculators were there. Then after that, computer came, software came, and now the third level, that is AI. AI is what? AI is nothing but next level of software programming. It is not that computer will be having intelligence. It is just the computer's software level will be at extra edge. So now here, because we all are pharmacy background persons, so here we will be discussing how we can apply that artificial intelligence in our experimental pharmacology, preclinical pharmacology, experimental physiology, how we can use those tools in our study, in our understanding. So by applying those tools, the software is designed which is called as XPharm. Another software is XPhysio. See, it is not so difficult. It is very easy to use. It helps you to perform the pharmacology experiment with the help of software. As you know, earlier animal experiments were being done, animal cruelty was there, but due to CPCSCA, now CCSCA that has been renamed, PCI and other regulatory guidelines, animal experimentation has been reduced. And now software is being used to perform pharmacology experiments. So this is the software which we usually use. So here the purpose of the session is to give brief idea what is the software, how to use the software and all. So see, in 1960, Prevention of Cruelty to Animal Act was passed. I think all of you taken your breakfast. Okay. So if I ask any question, you will be answering in between. Okay. Nice. So anyone from you have used software for pharmacology practical? Anyone from you have used till now? Anyone? Raise your hand. Anyone has used the software for performing pharmacology practicals? Okay, no issue. So software is there and that is being used. Why? Because animal cruelty reduction guidelines are there from different agencies. Like Prevention of Cruelty to Animal Act 1960 clearly states that wherever possible, the substitute should be used like model, films, chart, books, etc. So model, film, chart, book, etc. means because in 1960 software was not there. The act was passed in 1960. Today it is 2024. So at that moment they told that wherever possible, chart, films, these things should be used. But now because we are having latest technology, earlier we were saying software, but now in 2024 we will say AI software, artificial intelligence enabled software. So that is adhering to this 1960 Act. And to this Act adherence only Pharmacy Council of India, or you can say CCSCA, earlier it was CPCSCA. Do you have heard the name CPCSCA? Okay. So what is the new name of CPCSCA now? What was earlier the name of the committee was CPCSEA, and now the committee name is CCSEA. Okay. Committee for the purpose was there, so purpose word has been removed, otherwise it is same. So CCSEA, yeah. So CCSEA is also a body which controls the cruelty on animals. So anyhow, different agencies, Pharmacy Council of India, Ministry of Environment and Forest, CCSCA, all are working for prevention of cruelty on animals. Bureau for Health and Education is also, it's a philanthropic organization and basically works to provide education solution and also to reduce cruelty on animals. It has done MOU with PETA India also and various other agencies, including PCI, AICT. PCI has recommended this particular software in the syllabus. If you will see the B-Farm syllabus, the B-Farm syllabus, X-Farm name is mentioned. Similarly, in B-Farm also, they have mentioned that alternative software, alternative tools should be used. So this was the brief introduction. Now, what is virtual experimentation in pharmacology? Virtual experimentation in pharmacology means doing the same pharmacology experiment with the help of virtual techniques. Okay, I'll ask one question. There is one term called in silico. What is the meaning of in silico? Any one person? Anyone? Correct. But why we call that in silico? You are right. Your answer is correct. But now, how you will associate that silico word with software? From where this silico word has came? Correct. From silicon. You stand up. Then why? How you connect in silico with silicon? Because in the computer, you are very much right. Because in the computer, the chips are made up of silicon. You must have heard of Silicon Valley. Why we call Silicon Valley? Because there most of the computer engineers and IT companies are there. So silico word has came from computer chips. And in silico pharmacology, what is in silico pharmacology? Nothing but virtual pharmacology. So this can also be named as in silico pharmacology. So for in silico pharmacology study, we used to use the software XPharm. So this was the virtual experimentation in pharmacology is also called as in silico pharmacology. So Bureau for Health and Education Status Supplement is an organization which works for prevention of cruelty to animals to provide education solution. So from them, XPharm is also there, and it's not a one-day thing. Actually, it has evolved from many years, one by one. It's a you can say 20 years, approx 20 years old journey. The things which you are seeing now started 20 years ago in 2002 or 3 or 4. So one by one, different people came, different scientists came together on a banner, and then these things came. So what is virtual experimentation in pharmacology? See, now there are two things. In virtual experimentation in pharmacology, there are two things. One is software development and another is application. Because we are pharmacy students, so we don't have any role for the development. We are more interested for application of the tools. So application, if we know how to use the software, then we can say that we know in silico pharmacology or virtual pharmacology. So what is XPharm? As I already told you that this is a virtual pharmacology tool. With the help of the software, you can perform the experiments. And your institution is also having most of the institution actually through the university itself. Like yours is Nagarjuna University, one more is Anantapur University, JNTU Anantapur. So JNTU Anantapur, they themselves are taking the licenses from Bureau and then they are distributing to the colleges. They are giving the licenses. Yes, sir. So all colleges are using. There is nothing but a simple password is there. In Google, you have to write down XPharm login. Automatically, Bureau page will come. And there, when you will open that Bureau page, it will ask for ID and password. When you will enter your ID and password, all the module will open. And without the killing of animal, you will do the experiment. Like you will pick the animal, you will inject the drug, you will check the response, you will record, you will enter your name and you will print the result. So your complete practical will be in the form of system. If possible, I will try, otherwise you can later on practice also. And in YouTube also, all experiment videos have been given. When you will write XPharm, all videos are prepared by different experts also. Although I am principal mentor, but more than 100 pharmacologists are associated with that. From Andhra also many experts are there from various places. Medical colleges, faculties are there. So they have prepared videos also. So when you will in Google, you will write down XPharm videos, you will get all the videos. So this is the history, like how it has grown. As I already told you, like it is a 20-year-old thing, one by one, but today it is very much developed, up to AI level it is developed. See, when more than 5,000 college students are using, it means you can understand every day 20 to 25 lakh clicks are going on that. So you can understand how big will be the programming level. Because controlling the 25 lakh clicks every day is also a big. So these are the different experiment lists. Means at present it is having 87 group modules. So complete your B.Pharm, D.Pharm syllabus. Complete is there. PCI is also, Pharmacy Council of India has also stated. And in certain cases, they have told that if you are having the software, in certain cases, animal house is also not needed. So this is the notification. Similarly, ex-physio is there. Any PharmD student is there here? PharmD? Yeah. So in PharmD, do you have one experiment on gastrocnemius muscle? Do you have one experiment on gastrocnemius muscle? That experiment can be done because that has to be done on animal, and animal dissection is banned. So you can do that practical with the help of ex-physio software. This ex-physio software covers the need of medical also, pharmacy also. But in pharmacy, mainly this first section 1 to 7 is useful. Gastrocnemius muscle, neuromuscle experiment we usually call it, sciatic nerve and gastrocnemius muscle preparation experiments. So ex-physio, although ex-physio is not being used by many pharmacy colleges because in your syllabus PharmD only 1 or 2 practicals are there, but pharmacology you are having complete syllabus, so that is being used. But ex-physio also you can use for your PharmD course. So these are the different experiments. The purpose of showing experiment was like, because it has evolved from long duration, so almost all practicals will be there. Although few other, we can say organizations are coming with the, we can say their financial purpose and all, because in this case it is not a financial purpose, and the coverage is quite big. In this one, if you will add X-Pharm and X-PGO, more than 150 group module will be there, 150 practical, 150 group practical. So no pharmacology, physiology practical will remain. So once you will go through and once you will do all the things, if you are planning to become a toxicology scientist, if you are planning to become a pharmacological scientist, so all the practical application tool you will understand. Anyone can name any experiment of pharmacology? Anyone can raise the hand and can tell me. Tail flick, correct. Tail flick is there, rotor rod is there. So how you have performed that tail flick experiment in your laboratory? By using that one. Very good. So if you will say I have killed the animal, then you will be questioned how you have killed because you are not allowed to do. So to that level, this in silico pharmacology now has reached. You can get the software by filling the form. In Google, you can simply write down Bureau for Health and Education X-Pharm. You will get the form and everything. Already your institution is having, so each student need not to use that. They can directly take from their principal. And in Andhra, JNTU Anantapur is giving the passwords also. So if any institution is associated with JNTU Anantapur, they are directly collecting from there the password. JNTU Anantapur is directly having tie-up with Bureau for Health and Education. So advantages of virtual pharmacology. It avoids the use of animal, the most important. As you know, we have seen this pandemic, COVID pandemic. if you will see the basic reason behind that pandemic, that was nature, animal, and you can say human imbalance. Because if you will be killing the animals, you will be cutting the trees, nature imbalance will be there. Then these type of calamities will come. So if we will prevent the cruelty on animals, the calamities like COVID and all these will not happen. First, of course, we are preventing the cruelty on that. Second, you can perform many experiments in shorter time. Suppose if you want to do dog BP experiment, practically you have to bring dog. You have to cut the dog. Then you have to isolate the dog heart or you have to keep it in the table. Then you have to acclimatize that. It is a 3 to 4 days thing. And in through the software, you will do it in half an hour. You will open it, it will cut. You will understand the purpose is to understand the concept of different drug on the heart. So all the graphs you will get in the software on one click. And then you can interpret your result, you can write your name, you can print your data. So this is the short duration. Of course, the cost is also reduced for the institution. Otherwise, institution have to manage the big animal house and everything. In this case, they can simply take the password with a very economic costing. And test reports are, you can save your reports, you can print your report with your name. So the data will be more presentable and more easy to understand form. So these were the different, you can say, application of the software. And as you are saying, you have already used the software, so you can once again perform the practical. And in YouTube, when you will write down XFarm, you will get complete video series of all the modules. From there, you can understand, and then by clicking the software, you can open. Can we open the website here? We will try to open it. Do we have time? 5 minutes, sir? Okay, I'll show you the login page and the YouTube page only. Write down Google here. Google. Just 2 minutes, we will show them the page from where they can see the things and all. Because it's a big thing, it cannot be covered in the small section.
We are also discussing the same.
Yes, I agree. You can click here. See, in Google, you have written XFarm, automatically this has came. This page will came. Click here. Click here. Here. See, like that, it will ask you to allow the location. It will ask you to enter the password, and the complete software will open. It is very simple. You cross it now, and you open YouTube. YouTube open. Okay. Here in search, you write down XFarm. Okay, yeah, this one, first one, correct. On this one, you click this one. In playlist, you click playlist. Here, this one. Now you click on this one. So before performing, see all the modules, video, explanatory videos are. You can scroll these ones. All. See, all your BPharm practicals are there. XFarm series is there, and X Physio series is there. So first you can see the video, and through that login page you can easily perform the practice. And you will understand all the concept of pharmacology and physiology. Because when you are going for the interview, they will be asking from you the concept of toxicology, pharmacology. If you will do all these 150 group module, see at which level your understanding will be. So try to do all the practical. Okay. Thank you. So I would like to thank once again the management of PHITS. And of course, the session here and the dignitaries and my dear students, thanks for listening me, and we are always available. I am Dr. Dharmed Ahuja, means associated with the research principal mentor in Google. You can write down my name also, and anytime if any help is needed, you can get my number from your principal and all, or your management. The respective principal can also give you. So if any help is needed, we are always there. So thank you. Thanks a lot.
Well, thank you, Dr. Dharmed Ahuja sir, for your informative session on artificial intelligence role in drug discovery and development. So the talk was is very relevant to the theme of the international conference, that is accelerating drug discovery and drug development through artificial intelligence. So Sir has spoken about in vitro animal models and also in silico animal models, right? So which are alternative to animal research models. But as per the Animal Cruelty Preventive Act 1960, so they have asked to implement alternative models to on the animal research. From then, slowly, the institutes and research organizations, they are implementing the thing by including the alternative models. So also, as per PCI guidelines also, it has been made mandatory to use alternative softwares instead of animal experiments. But when we are studying, we used to handle the animal models, and also we used to do the like practicals on the animal models. But anyway, in order to bring about a drug molecule into the market, so one need to focus on the animal experimentation, because without animal testing, we cannot predict the toxicity and also pharmacology of drug molecules. So for that also some artificial intelligence models are there, like prediction of toxicity, prediction of pharmacological activity. So Sir has spoken about those talks also. So and also Sir has focused on regulatory agencies like CCCSA. So which is now it is renamed as CCSA. So Sir has given guidelines of animal models of CCSA also. So that was very informative session, sir. So from the audience side, so if you have any doubts, you can ask from the audience point of view. So do you have any doubts? Okay. So with this, I am going to conclude the session. So thank you very much, sir, for your informative session once again. And also, I thank the members of Chebrolu Hanumaiah Institute of Pharmaceutical Sciences and also members of Operant Pharmacy Federation for giving me opportunity as a chairperson for this session. Thank you, one and all. would like to, I would like to ask you a small question. So you said that the animal models which we are doing the pharmacology experiments, what is the translatability of the treatment? So, if we give, say, for example, 5 units of insulin to an animal model and we get the response, and the same thing we do it in the software, we get a response. So, what is the guarantee that both the responses that we get, the response that we got from the animal and the response that we got from the software, how can we assure that the response that you get from the software is definitely going to match with the response that you get with the animal model? Sorry, the live animal. So, is there anything that you can tell on that?
Very nice question. And you have asked very appropriate question like about the magnitude of the response. Actually, in the software, the responses are added based on if you will see the editorial board, it is having more than 100 pharmacologists. So nothing has been added without approval of all the pharmacologists. So due to which you are having the advantage that all the results and everything are at an accurate level, because it is not like one company, small company, one pharmacologist is coming. In this 100 pharmacologists, when they all will approve, then only that result will be added into that panel. And as I told, it is not a one-day thing, it is a 15-20 years journey. So it has refined to a very high level, because see, as you ask questions, similarly, 5,000 colleges, India, abroad, they are also asking questions. And every day, refinement, and due to the cloud base, if anything is there, any update is needed, we used to do at the same moment. So that's why that is one more advantage, you will get the accurate understanding of the topic. Otherwise, sometimes there could be misunderstanding of the topic. So that will also not happen in this. Very nice.
In case if we identify any toxicological response in the software model itself, are there any other software programs that tell us how we can modify the toxicological effect?
See, this is for learning purpose. This means already predefined data is there in this one. So when you are going for the research part, then you have to take animal only. Suppose you are doing PhD research, then you have to take animal only. But when you are doing at M-Pharm level or at B-Pharm level, there you need not to find out the unknown sample effect. So in that case, you can use the software because predefined data is there. But when suppose you are going to identify any plant extract effect, then you cannot go for the software. Like it is not like you will put that plant extract in the software and it will give you result. It is not like that. It is for computer assisted learning, or you can say animal simulated learning, or you can say in silico pharmacology, or you can rename virtual pharmacology. But to that extent only, to a predefined data presentation and understandability at an accurate level. For this one, it is once you will understand all the things, then when you will go on a research environment, you will have a confidence to do the things. For this purpose, it is.
Thank you, sir.
So with this, we came to the end of plenary session 3. In this moment, I request our principal and the organizing secretary of the conference to please come onto the dais and felicitate the speaker and the chairperson. At the outset, I request our principal and the organizing secretary to felicitate the speaker, Dr. Dharmendra Ahuja. Now, I also request the speaker to please join the felicitation for the chairpersons. And first, I request Dr. D. Narendra to please come forward and accept the felicitation. Next, I request Dr. T. Vinay Kumar to please come forward and accept the felicitation. I request all the audience to stay in the room. We will start plenary session 4 within 5 minutes.
millions of dollars in a drug design itself. So after that, if failures means total millions of dollars are wasted. So the previously what the traditional method of drug design is highly risky one, because after we are preparing the drugs, it may be failed to produce the activity, or it may produce some side effects or adverse effect. It is totally waste. And so it is a long time, more than 10 years it required to synthesis, and it comes to the market. From the designing to come to the market, minimum 10 years are required to produce a single drug. So that is the disadvantage, and it is more expensive. So millions of dollars they can spend to produce a single drug. And the drug is highly regulated by Food and Drug Administration, FDA approval. Getting FDA approval itself very difficult. So after you are completing everything, you can't get approval, the total process is wasted. And the investors, those who are investing to discover new drugs, they want to some outcome. So this is there are 4 main phases are involved. So one is first discovery of drug. First discovery of drug using computational based or any other manually you can discover. Then development. So after discovery of drugs, we have to develop. So some modification we required to meet all the admin properties, everything. And then only it goes to clinical trials and finally comes to the market. So there are 4 stages are involved in the drug design. So this is normal drug design. So first we have to identify the target. So which target you want to focus. It may be a cancer or SARS-CoV-2 or any other targets. So that first we have to identify the target, then validate the target. So what are the binding pointers? So target means this is nothing but a protein. Simple protein, it is made up of a lot of amino acids. So any drugs that can be bind with that particular protein, it may be inhibit or it is enhance the protein and produce the activity. So that is the common mechanism involved in the drug discovery. So then from this target, so we have to identify some hits. Hits means, so what are the compounds which produce activity against that particular targets? So that is called as hit molecules. So from this, so we will select 1 lakh molecules together, and we will check the activity by using computational tools. There are a lot of tools are available. So from that we can identify only 100 molecules, 100 top active molecules. Then we'll go for the lead. Lead means which is the top active and which is able to produce activity, and you have to check not only some compounds may produce activity, but it fails in pharmacokinetic characters. Then that also totally waste. Sometimes not absorbing in our body. So we have to see each and everything. Not only able to produce activity, it possesses all the admin properties. Admin, absorption, distribution, metabolism, excretion, and toxicity. So that profile also pass, then only it goes to the market. Then only we are able to get FDA approval. Then, so for this, for each stage, they have to spend a lot of millions. So nearly 94 million for mid identification itself from any company. Then 166 million to identify the lead molecules. Then lead optimization. So we have identified the lead which is more active against the particular target. Then optimize that lead. So we have to modify slightly. If any failures in absorption means, we can slightly modify the drugs to increase the absorption or metabolism something. Then lead optimization. Then only it goes to the preclinical stage. So you have to test first in vitro analysis, then in vivo by animal models, and finally it goes to phase 1, phase 2, phase 3 clinical trials. After completing all phases, then only it comes to the market. We are going for approval, FDA approval. After FDA approval, it will come to the market. So generally this process will take more than 13 years for a single drug. And sometimes so you will complete all the stages, but we have to see simultaneously the other properties also. So bioavailability, admin properties, toxicity, and selectivity and metabolism, everything we have to see. Not only it produce activities, it is a good drug. So it will pass all other processes also. So if it is produce activity, this is a more potent drug. But it does not able to absorb by our body, that is totally waste. So they are spending this much money, that is totally waste. Billions of dollars they have to spend. So this is the traditional method. So we have to check each and everything, all the property gives pass, then only it goes to the market. So this is general drug discovery. First, disease target hypothesis. First, we have to analyze the target. So what are the proteins, what are the amino acids are there, what are the binding pockets are available. So where it can bind the drug. Then if you select 1,000 from the 1 lakh molecules, only 1,000 molecules pass that initial stage. Then only go to assay and screening. Screening with the different now ready-made screening also available for different topics. It may be cancer or any other disease. So we have to screening that drug. Out of 1,000 only 100 molecules will be passed that screening. Then finally, out of 100 only 10 molecules are passed the other parameters. Many medicinal chemistry and pharmacokinetic activity, admin properties, everything will pass only 10 molecules. Finally, all stages is over, only one molecule. Out of 1 lakh molecule, we'll get only one molecule as more active. Okay, this will take more than 10 to 13 years to get a single drug. So we have a lot of sources available. So we know how to use that source. So see this example, a lot of data are there. So he don't know how to climb that one. So the single ladder is sufficient. So how to use that sources? A lot of sources are available. If you check in the internet, they will put one anti-cancer drug means there are lakhs of cancer drugs. They are reported already. So from that also you can select, and from that you convert it into more active lead. So slight modification is required. So there are 3 phases are involved. First we have to develop the idea of the, so first we get some idea about the combo and idea about the disease. Then it goes to the innovation. So how to modify the drug into the active compound. And finally the implementation of that particular project. So there are, what are the impact of new drug discovery? So old drug discovery already we'll see. So there are more than 10 to 13 years they have to spend to produce a single drug by the industry. And also they have to spend billions of dollars to produce a single drug. So then only it comes to the market. After come to the market also, some drugs are failure. Failure, it will fail to produce any absorption, any failure in admin properties or pharmacokinetic properties, something is required. So in 21st century only we are introducing the new era of computational method. So what are the advantages? So previously they are spending 13 years. Out of this, 6 years they are required to develop a drug. Before synthesis, they have to develop a single drug itself. More than 6 years it will take. So they said that time spending we have to reduce by using computational method. So what are the advantages of computational method? So we have to develop millions of drugs, and already existing drugs also we can take from FDA approved drugs. So a lot of databases are available in the website. From that we can download millions of compounds together. Then it will, by using the AI method or computational method, first we have to identify the activity. So initial screening we have to do. So already existing drug also we have to modify and slightly. So during COVID time it is a very difficult situation throughout the world. So a lot of people have died due to COVID. So they don't know what. So for initial, we have to produce a single drug for COVID. It takes more than 10 years. But until 10 years we are waiting. Lot of persons we lost. So for that time, the computational method is highly useful to produce a repurposing. That is called as drug repurposing. So already existing drugs, we can use it for other disease. So that can be possible by only the computational methods. So we'll take lakhs of compounds, existing compounds, which is approved by FDA. From that, we'll check the activity against the other drugs. So already antiviral drugs are mainly used for COVID that time. Okay, antiviral drug, anti-malarial drugs. So many drugs we are used. That can be possible only in the computational system. So we can't directly will take and give to patient and test. It is not possible. So initially we have to design or slightly modify that compound. Initially we have to check with the computational method. Okay, whether it is active or not. Then FDA approved drug means maximum it passes all the pharmacokinetic characters. So that is not a problem for us to use. So only we have to check whether it is active against COVID. So anti-malarial drug, hydroxychloroquine, they are initially used for COVID treatment. So that time, simply they have to check with the computational method. Then if it is active, they will give it to patient. So that already established a drug. So there is not a problem to use by human. So that is the advantage. So there are a lot of studies now came about the protein. So genomics, we have to study about a lot of new courses also come like genomics, bioinformatics. So we have to identify more protein structures. So proteins are available in the website itself. So there are some databases available for the PDB. So high throughput screening means, so we have to screen lakhs of compounds together. So out of this, we will select which is the top active compound. And virtual screening, so molecular docking. Molecular docking means, so that is we have the target, we have the get, both together, we have to bind together. So which is produce more active. So more binding affinity is there, that is more active. So that is molecular docking. And the combinatorial chemistry means, so we have to simultaneously we will synthesize or design lot of compounds by changing small substructures. So that is computational or combinatorial chemistry. And in silico automatic screening also we have to determine by computational software itself. So automatic screening we can't check each and everything directly. So initially we have to check by using ADMET properties. So ADMET properties will check all the parameters simultaneously within few seconds we can identify. So what are the absorption, what is the dipole moment and molecular weight, everything. So log P value, pKa value, everything we have to identify by the software itself. So this is the recent after computational chemistry will come. So first we have to identify the disease and isolate the protein which is responsible for the particular disease. Then by using the genomics, proteomics and bioformulation methods, so we will first we have to study about the target. So what are the amino acids are there? Study about the protein that can be studied by the genomics, proteomics. Lot of courses are available now. Then both together we can know means high throughput scale. So we will download lakhs of compounds together from the existing compounds, or we have to design new compounds also. Then we will go for virtual screening by computational method. What are the compounds are more active against that particular target? So now the target specific treatment is very important. So previously cancer means will give general drug. So the cancer itself, there are a lot of different types of cancers are available. Liver cancer, breast cancer, lung cancer. So we don't know. So in that particular cancer, so some breast cancer is there, that also a lot of different targets are available. So we have to identify that particular target. So we'll give some other drug for general means a lot of things not active against that particular target. For example, breast cancer. Breast cancer, there are only ER positive breast cancer is there, estrogen receptor, that is one of the target. And HER2, human epithelial receptor, that is another target. And some progesterone receptor is there. Some breast cancer does not have all these three targets. That is called as triple negative breast cancer. If triple negative is there, we will give the treatment for that ER alpha means totally waste. So what type of cancer we have and particular target specific is very important nowadays. Then only it can be curable as early. And finally, so after virtual screening, we will design some different compounds by combinatorial chemistry method, then by molecular modeling and molecular docking. Then finally, you will select the top 10 molecules that we have to synthesize. Instead of synthesizing 100 molecules together, 100 molecules you want to synthesize means, so there are more than 2 to 3 years it will take. So after synthesis, all the 100 molecules may not be active that much. So for this, the advantage is first we have to design all the compounds. We'll check preliminary all the studies, all the parameters, physicochemical parameters, and all the activity, biological activity, target, which target it can be active. Everything we have to study theoretically by using computational tools. From that, we'll select the top 10 molecules for the next wet lab. So we have to synthesize only top 10 molecules, which that is more than 90% is active because already we did all the preliminary studies theoretically. So these are the theoretical method only. So after practical proven only it is accepted. Then after synthesis, you have to check the in vitro, then in vivo, finally it goes for clinical trials. What is the goal for drug discovery by computational method? So you have to identify the new molecules or existing molecules for different targets. That also we have to do. So number of compounds tested simultaneously for different biological activity by using AI method. Now artificial intelligence, the automatic software are there. So to give the input means it can analyze all the parameters on all the targets. In which target it is more active, it shows. So we can separate easily. From that, we have to select some molecules. and increase the number of protein targets. Each and every day, so new drugs, new diseases are coming. So we have to identify the new target. For that target, they have to isolate the protein, particular protein, and they have to deposit it in the website. So it is easily, freely available for each one. Each one we can download directly from the website. That is there are 2-3 websites are the RCS, BNC, VI. That website we can download directly the particular target. So that target, so protein target we have to download. Then what are the advantages? It is a simplicity. So first it is very simple. So no need to only one time you have to spend for software. A lot of free software are also available. So directly we can download the drug or we design the drug, and within few days we can design more active drug for particular disease. And rapidness. So rapidness is very important. So previously they are spending for designing and development more than 3 to 6 years. Now within 3 months we can complete everything. All the preliminary studies we can design, and final molecules we will select. And the cost is low. For synthesis of 100 compounds, how many members are spending and how much chemicals we are wasting. So this is not required for in the computation method. So only we have to download, everything is available. Directly we can download all the molecules, existing molecules, or we will design some thousands of new molecules. New molecules are just we can change. If we instead of chlorine, we'll put fluorine in one molecule and 2 chlorine, trichlorine. So simply we'll change the some functional group. So we'll get lot of molecules simultaneously. So already we know existing molecule for example, paracetamol, simple molecule that is for fever. So you want to improve the more potency. So there is some liver toxicity is there for paracetamol. If you are using continuously. So to avoid this, so slightly we change the molecule. So what are the important group? Already we keep as existing. So we'll put some new groups in other places, and we'll check the activity by using computational method. If you get very good drug, a single drug also is more successful. And higher information. So once you will keep in computational means, so that will be available there itself. So a lot of databases are available. So number of harvesting. So we can download, anybody can download throughout the world. Throughout the world, what are the scientists, they are reporting different molecules, we can extract from that easily. So that is information should be more. Previously, we are doing only manually. So you have to draw by pen or camera, and they will take picture and keep in our project previously. Now computation means it should be saved there itself. So anybody can download anytime. And the high efficiency compared to the previous traditional method, it is highly efficient. And minimal human involvement. So previously there are 3 years means how many persons are working for 3 years, nearly more than 1 lakh days they are working. 1 lakh days for how much money we have to pay for number of humans. Here only one person is sufficient to do all the work. By using only one system, one person is sufficient. The technical person, they can download everything, they will do all things within the few days. We can design a new molecules. And the computer drug design, so applications already I told. So there are a lot of molecules we have to check with the activity. And so macromolecules is nothing but a protein. So protein and amino acids, that is called as macromolecules. And small molecules are our ligands or drugs. And the pharmacophore is a common features. So all the existing drugs we have to collect already reported and proven activity drugs. We have to collect 1,000 molecules. From that, we have to search what is the common features for all the 1,000 molecules. That manually it is not possible. So we'll give the input in the system, automatically it will give. So one ring system is required, one hydrogen bond donor required, one hydrogen bond acceptor required, and what are the other parameters? Common features, that is called as pharmacophore. So we know the pharmacophore means, based on that we will design some new drugs. And it can be suggested in which place, what are the new groups are included, how to improve the activities. So existing drug also, we have to, based on pharmacophore, will improve the activity by introducing some new groups. And so structure activity relationship also very easy by using this. So there are 2 types of computational method is there. One is ligand based, another one is structure based. Ligand based means already I told pharmacophore. So ligands means molecules. So we know the molecule for a particular drug. So we know the already existing or 10 molecules are already our FDA approved. So we'll take all the 10 molecules, we'll take common features for all the 10 molecules. Based on that, we'll design some new molecules. That is ligand based. Structure based means this is based on target. So target we know. Target means cancer target. Cancer, also particular cancer, breast cancer. In this breast cancer, we want to target only estrogen receptor. So we'll take that target. We know the target, what are the binding pocket is available. Based on that, we'll search the molecules. What are the molecules which is active against that particular binding pocket? So that is same only target centric and compound centric. Target centric, that is a structure based medicine molecular docking. So docking, how you know? The docking based on score only we have to do. So some output we record, which is more active, which is less active. So there are one glade score. That is, for example, Schrodinger we are using, we'll get the glade score. Similarly, a lot of software, there are different scores are there. So based on the score, what are the top score we have to select. So by scoring function, we have to select. And de novo design means based on that, we will design some new molecules. And ligand based, similarity searching, similarity already, what are the molecules similar to that existing molecule, we have to search. So some slight modification here and there will be there. But common structure should be similar. So that based on that, we have to design some new molecules. So this is structure based drug design. It is easy and we are doing regularly structure based drug design. So it is a just lock and key model. So we know. So particular key only is able to open that particular lock. So similar way only, that is mechanism is similar to that. So key is our ligand. So lock is the binding pocket which is present in the protein or target which is responsible for the particular disease. So which is perfectly fit to that key and lock, then only this can be opened. Similarly, which is perfectly that binding pocket is there, which binding pocket perfectly fit with particular ligand, then only it will produce more active. So that same model only. So that binding should be possible here with lot of bonds, some temporary bonds, hydrogen bond is there. Because all the amino acids, we have the amino acid is in the target. So amino acid means, so amino group, acid group is there compulsory. So it is easy to produce hydrogen bonding. Hydrogen is available in both acid group as well as amino group. So hydrogen bonding with ligand. So ligand have OH group or ligand have OH group. It may be form hydrogen bond with that NH2 group. So it is tightly binding. So similarly, lot of other bonds, covalent bond and other polar molecules or different bonds are available. If more tightly binding, which is perfectly fit to that particular binding pocket, then only it is able to produce more active. So docking is a identification of low energy conformation. So in the docking itself, there are a lot of possible. So one drug is that it bind with that pocket. So it may be binding like this. There are different conformations. So which is low energy conformation, that is more stable and it is bind perfectly. So based on that, we have to see this lock and key model. So this is only perfectly fit to that particular block, then only it can be opened. Similarly, so this is the binding pocket. So the green color is this is binding pocket. So this molecule should be perfectly fit to in that pocket only produce more active. Okay, so that's same mechanism only. So simply you have to understand. So docking, first you have to identify the target, receptor. So this target, this is a binding point. This hole will be available in the protein itself. So every protein has some gap is there. So that gap only we have to fill with our molecule. Then only it will produce active. So we have to decide some ligands. So there are a lot of ligands are there. So which ligand is perfectly fit to that pocket? So out of this, so only this is similar to that binding pocket. So that ligand only it is bind to that particular receptor. So this also there are different direction can be fit. So which direction is perfectly fit, that will produce more active. Okay, so that is a conformation. So best conformation, which is the best conformation that will produce more active. So for your easy to understand, you will see this how it acts.
Video. Next. In this video, to easy to understand. Particular protein, so the large molecule, that is a macromolecule, that is a protein, this is a ligand. So first you will find the binding pocket, which is perfectly fit to that will produce more active. So it will go and search the binding pocket. So this is a lot of macromolecules. Mainly binding is due to that bonds, hydrogen bonds. It's a temporary bond is required. If it is a permanent bond, that is a problem. It's not excreted easily. So mainly temporary bond like hydrogen bond or hydrophobic interaction. Hydrophobic between lipid because maximum of cells are made up of lipids. So lipid interaction. So hydrophobic in the hydrogen bond, it will produce more active. So this is another different types of pi interaction is there and pi stacking. So pi stacking means so that aromatic molecule should be between two aromatic compounds. So a lot of aromatic amino acids are also available. So between this, it will produce more active. So let's see it again. So drug first we will go to that protein and it finds the binding pocket, which is the binding pocket, which is the gap in the protein is there. They are able to find out that protein and perfectly fit to into that. So that fitting is mainly due to some temporary bonds. So it may be pi stacking or hydrogen bond interactions or other interaction, temporary interaction. If it is covalent bond, it's very difficult. It is not breaking. So it is permanently in our body itself. It is not excreted easily. That also creates a problem for us. So this mainly hydrogen bond plays a major role because amino acids have amino group and acid group that easily can form a hydrogen bond. So our compounds also have some hydrogen bonding related to any hydrogens. Many hydrogens are there. If OH group or NH group is there, that easily form hydrogen bond with that particular amino acids. So there are a lot of docking softwares are available. So we'll see this is the So these are the few docking softwares. This is freely available for the academic purpose. The Dow, Control, Surplus Spread, and then commercially available is Gold, Glide, G Docker. So these are all commercially available software.
What is the advantage for commercially available? There are a lot of parameters. That is very more accurate and a lot of parameters are available in that. But it is too much costly. So we have this Schrodinger software in our university that is spending that full version. We are spending more than 3 crores for 5 years. But so we have to purchase only few modules also. That is cheaper. So what is required for us? Based on that, we can purchase. So there are different stages for docking. So first we have to identify the receptor of target selection. So we have to download different targets. Target means nothing but protein. So we have to download the target. Protein is available in various databases are available, freely available. So we can download any target protein for the particular disease. So always that 3D structure we have to deposit. So we download that particular protein. We can't use as such. So first we have to prepare the protein. Prepare protein by using some software modules of that specific modules. We will see in the details in the next one. And identify the active site. So what is the active site? Binding pocket. That is called as binding pocket. They are only our drugs to bind and produce activity. Then ligand selection and preparation. So we have to select ligand. So ligands also a lot of databases are available. So there are millions of compounds, existing compounds are available, and we have to decide some new compounds also. For our requirement, we have to decide some new compounds. Then to go for the docking. So docking is nothing but so we have to work together. So prepared ligand, prepared protein, both react together. So it will identify the binding pocket and perfectly fit to that binding pocket and produce activity. And based on the scoring function, we have to select which is the best model. Okay. So first one by one we'll see in detail. First receptor selection. So proteins, already I told proteins are available in the website, freely available. So anybody can download. So we know the protein PDB code, some PDB ID is there. So if you know the ID, so we can see from the literature also. And directly you have to search that ID. You can get, first you have to identify, there are two major databases available. One is NCBI, that is National Center for Biotechnology Information, that is US based, that is ncba.nlm.gov. And another one is RCSP, that is Research Collaborative for Structural Bioinformatics. So this two is the major database for target selection. So if you have to identify any target, you can download directly from this 3D structure of that particular protein. Then that 3D structure we have to, we can't use as such that one. So we have to prepare that protein. Because the protein, some proteins have 500 to 1000 amino acids, up to 1000 amino acids. So all is not required for us. So if you take all together means it will take more time in the software. So you have to decide some grid. So only that particular where the binding pocket is there, that area only we have to select. Only 10 Armstrong. That area only will select it. So only that particular area is required for us to produce the activity. So here we are using stored in the software protein preparation resort. That one option is there. That is a EPIC model. So that we have to use for protein preparation. So this is the protein. Protein is like that. You download any protein. So this that is a cancer protein. This is SARS-CoV-2 protein. This is ID. So protein ID so 6xB1. That is what we use. That is 4-letter code. Generally, PDB ID is 4-letter code. Then receptor grid generation. You have to select that particular area of binding pocket where the binding pocket is there. So there is a large protein. So you have to select that grid. The grid is only 10 Armstrong to 10 Armstrong. Where the binding pocket, only that area we have to select for the docking studies. Then ligand preparation. Ligand also we can't use as such. Because if it is 3D ligand only, it is flexible. So to identify the binding pocket and perfectly fit with that, there are different orientations to be found. If 2D structure means nothing we can't do. So first we have to download the 2D structure of ligands. It may be existing ligand from the database, or we have to decide. Some will draw by using simple softwares. The ChemOffice, ChemDraw, many drawing softwares are there. So we have to draw that software and we'll keep ready. Then it converted into 3D structure. Then only it become flexible. So that can be always we'll do the flexible docking. So perfect in 2D means only one direction we can dock. It's not perfectly fit with that target binding pocket. So 3D means it is flexible. So 3D structure we have to convert it into 3D structure. And by using that here, liquid module is there. In storage software, liquid one module that we are using for the protein preparation. So after preparing protein, we have to minimize the energy. If always low energy conformation, so there are a lot of conformation is there. Same drug, there are different directions. It is 3D structure. There are thousands of conformations for the each combo. So you have to select the best orientation, best conformation with lowest energy. So for that, we have to minimize the energy by using some energy minimization force field. Here, OPLS force field we are using. OPLS means optimum performance liquid simulation. So that we have to use to minimize the energy. Always lowest energy is more stable. So that combo, that energy conformation we have to select for docking. So already we have the prepare the protein, prepare the ligand, then we'll go for the docking studies. So docking is, so there is a model, glide model in storage assured. So a lot of the auto dock and others also available, but we can't get more number of parameters in that. So we are using storage software that is a glide model is used for docking, glide docking. So in this software will select the ligands, select the prepared protein, prepared ligands, not as such we can't use. So we have to prepare the ligand, prepare the protein by using different modules, then we'll combine together. So make us a docking studies. So docking studies, what is the output will come, how to analyze the docking. So there is some score will become docking score or glide score. Here it is called as glide score. Always will get in the negative value. So if it is more negative value, it's more tightly bind to that particular protein. So that is more active. So based on that, we have to select the compound. So here also there are different modules. In this module, there are different methods are available. So there are HTVS, the high throughput visual screening. So that how much time or how much accurate based on that. If you have more than 1 lakh molecules, you want to do simultaneously documents, you have to select that one, HTVS. Only few seconds for each molecule, it will spend only few seconds. So that is not accurate. For initial screening, we can use HTVS, high throughput visual screening. So 1 lakh molecules you have to select. All together will put in a particular target protein. So from that we can select only top 100 molecules for the next stage. The next method is SP method. HTVS, SP means standard precision method. So that also not that much accurate, but compared to HTVS, this is more accurate. If 100 to 200 molecules, you can use that one. So for that molecule spend more time on that particular protein. And finally, XP method, extra precision method, that is a more standard. So only few molecules, but it will take more time. So how much time it spends to the molecule into the target, then only to identify the perfect binding pocket. So it takes more time. For each molecule, it will take more than 5 to 10 minutes based on your speed of your computer. Extra precision mode. So based on the favorable, there are a lot of parameters we have to analyze. So some hydrophobic or hydrogen bonding, and some there are already we will see that video that only tightly bind the ligand with the target. So based on that, we'll get the score. Based on score, we have to select the top active molecules. So first 1 lakh molecule select, go to HTVS, high throughput visual screening. From that, top 100 molecules will select, go to SP method. So we have to save the time. So all the 1 lakh molecule will put in XP method means it will take more than 2 months to complete based on your system speed. And finally, so not only activity, it will produce activity itself. It is there is no use. It is produce more active, but it failure in admit properties means that drug is not a good drug. So druggable, whether it is druggable or not, the compound can be identified by admit prediction. So absorption, distribution, metabolism, excretion, and toxicity. So all parameters it will pass, then only it will be a good drug, not only based on the activity. So this is the docking. So docking are, for example, some 20 to 30 molecules docked together. So all are between the particular binding pocket only. This is the protein. So these are different amino acids. So valent 20. So each protein have, each amino acid have one number. So the 3 in 21, spot in 28 like that. So how many number of amino acids are available? Based on that, there are different proteins. They have the particular binding pocket. That binding pocket only all the drugs are attached. You will see all the drugs are aligned only in that particular binding pocket. So this is a structure based. Structure based is very important and easy to identify the drugs because we have to use lots of drugs together. And another one type is ligand based. Ligand based, we know the ligand. Already I told one activity, one active ligand. Already it's reported for particular disease. So for that, already we know the. I told next on paracetamol. So paracetamol for fever. Already it is very established drug. So you want to produce new drugs for fever means that take as a standard. So slightly modify in that. So that is called as ligand based. Ligand we know, but target we don't know. So based on the ligand, we have to modify slightly, then we check the activity. That is a ligand based. So mainly pharmacophore. So we'll take 100 molecules under the active reported molecules will take 100. So we'll take the which is the common parameters, common features for all the 100 molecules. Based on that, we can select the top active molecules. So we'll design new molecules based on that. So mainly that hydrogen bond donor, how many is there? So this is the common pharmacophore model, ADHL. So AD means one aromatic compound, so acceptor, hydrogen bond acceptor. D means hydrogen bond donor. And hydrogen means hydrogen bond donor, hydrophobic. Hydrogen means hydrophobic. So maximum molecule should be hydrophobic. Then only it is able to cross the blood-brain barrier and lipid. Our cells are made up of lipids. So next we will see one example. So how we are going for talking and how the result we can analyze. So case study. So already we did for breast cancer. So breast cancer already you know. So there are a lot of breast cancer, you know, mainly it is for many of the people more than 40 years. They are affected. Almost 5:1 people can be affected with breast cancer. So that is very important. There are a lot of targets are available for breast cancer. There are different targets, luminal A, B. There are different mechanisms are involved. So luminal A can consist of only two targets: estrogen receptor and progesterone receptor. We have estrogen, progesterone, and HER2. It may be present or absent. And C, another one, HER2 expressed. There are different types of breast cancer is there. So in this, which type of breast cancer we have, based on that only we take the drug. So for some breast cancer does not contain all the, so more than 50 to 60% is mainly ER positive and progesterone positive. More than 60%. Only that 20% is ER and HER. And the remaining 15%, that is only very dangerous. So this one, generally we have this progesterone and estrogen receptor. That receptor, that is common. Some triple negative breast cancer means all three targets are not available. It may be some other target. But we are giving drugs for this target means there is no use for that, not able to cure that type of breast cancer. Okay, so we have to know first which type of breast cancer is there. So based on that, we'll decide the drug individually. We say that particular target, which target we have to inhibit, that target based we have to decide the drug. Then only it is active against that cancer. Simply we'll give some cancer drug, it does not work for this particular activity. So we have to decide some normal compound. So this is the workflow. First we have to find out the target, which target we want to do. Okay. Then go for the virtual screening. Virtual screening means all the 1 lakh molecules we can collect. Then go for the, there are a lot of the packaging and these are the databases. Protein also we have to download from database only. That already I told RCSB and NCBA. We'll download the protein. And the drugs also we have to download with the different databases. That is zinc means that consists of 1 million drugs. Like that Campbell and PubChem. So there are a lot of data, free also available, paid also available. So we can download simultaneously 1 lakh molecule or 5 lakhs molecules. From that, First, all the 5 lakhs molecules will, we can't do the docking, it will take more time. First, we have to filter by using some online tools. How many molecules are drug-like properties? And how many molecules have toxicity? So based on that, we have to filter one by one. First, online tools, by using a lot of tools are available. Based on that, we have to filter some molecules. From 5 lakhs molecules, we can filter, after filtration, we'll select only 50,000 to 60,000 molecules. Then it goes to docking. First TBS, then SP docking as a standard precision mode. From 50,000, it goes to only 500 molecules to standard precision mode. Then that 500 molecules subjected to extra precision mode, we select top 50 molecules. That only will give full docking studies. Okay, first docking study of that 50 molecules, we take the top active molecule. And also simultaneously, we have to check ADMET properties also. Because it produce activity, it fail in ADMET means it is not a druggable. ADMET properties, then we have to analyze after docking, that is whether the docking should be stable or not. Some drugs only few minutes it is there. So it is moving only. So it is moving and immediately it is coming out from the binding pocket also possible. So how much the docking complex should be stable? That can be analyzed by the MMGBSA. That is post-dock analysis. So molecular mechanics, generalized bond surface area. That is another model. So after docking, we have to analyze how much it is stable. So that stability can be, that is binding free energy calculation. So how much it is stable, that can be analyzed by MMGBSA model. That is a prime model available in the Chowdavaram. Then finally, top active molecule, which is more stable, it will produce more docking score as well as that delta G, DG bind value. So that is a binding free energy calculation. If both it is good, that molecule will check for molecular dynamic simulation. Dynamics means how it acts in our body. So in our body, not only the protein. So we have a lot of more than 70%, we have the water molecule. So in the water molecules and other present in our body, how much it is stable, how much it is active in our body, that can be analyzed by molecular dynamic simulation studies, MD simulation. That is Desmond model. That is another model, Desmond. That is used for molecular dynamic simulation. So after that, all only it goes to the next stage for wet lab. So first molecule docking already explained. So we have to take all three. So 2 IOG is a ER pop target for breast cancer, and this 3BP0 is HER2, and 4 triple Z is PARP1, that is triple negative. So take all three targets simultaneously. We'll design some compounds, 20 to 25 compounds will design. So we'll check for all three targets, which is these molecules are active, which is inactive. So based on that, then we'll go for the ADMET prediction by using prop model. And MMGBSA, after docking, you have to check the binding free energy calculation and finally molecular dynamic studies. So all together we have to do. So this is the result. So we design this many molecules. So we are designing some acrylic derivatives, which also connects diacetylates. So nearly 24 molecules we are selected. We are designing new molecules. Or we have to download from the existing also. So this is the plate score. So we get. Or for comparison, we will take some standard also. Tamoxifen is a standard for breast cancer, ER. So we'll take one standard from the breast cancer and another standard for acrylic derivative, which is available in the market. That is used for cancer. So there are two standards compared to these two standards. So these are the top 10 molecules. This is more. You see, -11 is a standard. So all the compounds have more than -11. So compared to this one, our molecule should be more active. Plate score -12. This is top active molecule. So this score is consist of, it is a all together of other parameters, lipophilic and hydrogen bond. So there are different parameters of that. So this is score is combination of all the parameters, some positive parameters, some negative parameters. Sometimes it will have good score, good hydrogen bond, but it has some negative parameters also. So due to low molecular, there are some rotational penalty. So penalty means a lot of single bond means it is able to rotate. So due to that, the score will be reduced. So all the negative score is positive parameter for docking score, or the positive score is a negative parameters. So this is the combination of all together, range score. So we will take only that range score. And based on that, if any rotational penalty is there, so for say some counter minus plus 0.17. Rotational penalty means single bond. Some single bond it is rotated. So we have to avoid that one. That bond is converted into double bond, or so we have to attach some molecule there, some functional group. Then we have to reduce, we will improve. So based on each and everything, we can modify slightly to improve the activity further. So these are the talking result that 2D structure is common. This is the 3D structure. It comes like that. This is the binding pocket. So this is the 2D interaction structure. So where the interaction is there. So these are the binding pocket containing amino acids. In this area, there that arrow. See that violet color arrow. So that is a hydrogen bond. So mainly hydrogen bond that acrylic have hydrogen bond with aspartate 363, 863. So that is important amino acid that only interact with the ligand. Similarly, this also aspartate 800 have hydrogen bond with that NH2 group. So that two amino acids are very important for our binding pocket. So due to this hydrogen bond, it will produce more active. So based on that, we have to analyze each and everything. So this is for other two targets. So same drugs with other targets. So this is the against HCl2 3PPO. This target. So compared to this one, already having less score, but it does not active against HCl. Only minimum activity is there. Similarly, the triple negative, you see that also very less active. So our drugs are only suitable for ER alpha targets. So simultaneously we will analyze all the targets. What if one compound have inhibit all the three targets, that is very good compound. So we can use it for common all the type of breast cancers. So this is for HCl2. Here are two. Here there is no any hydrogen bond. So this score is very less. So this is for part one. Here some hydrogen bond is formed. There are three hydrogen bonds are there, but score is less, not that much active. So this is the result for MMGPSA, binding free energy. So this also total of all the parameters like Coulomb energy, hydrogen bond energy, lipophilic energy, van der Waals energy. So what are the energies are help to increase the binding energy. So binding, tightly binding means it is more active. So we get these are the TG total value of the MMGPSA. So here minimum minus 59 to minus 80. Again. So this is the best molecule. Compared to this one, this is the best molecule because it is the stability should be more. But that score wise, it is slightly less. Instead of minus 12, it may be minus 11.5. But compared to the top part of the molecule, the stability of one E should be more. So we'll select this molecule. So it will produce more active as well as the stability of docking complex also more. So based on that, we'll select the molecules. And then these are the admit properties. How the result will come in admit properties. So number of donor hydrogen bond, acceptor hydrogen bond, log P value, number of metabolites. So KHM means protein binding value, and the rule of Lipinski's rule of 5. You know. So how much deviation from the rule of 5, and how much overall absorption. So all the recommended value we have. So all are within the recommended value means it is a good drug. It is a more useful drug. There is a pass all the admit parameters. Not only it will produce activity, it will also pass the all the admit parameters. Then only it is a very good drug. And then final compound. So only top active compound will go for detailed study. So how it behaves in our body. So based on that, so molecular dynamic simulation. So dynamic simulation, the DESPON module we are used. So 100 nanoseconds will run that dynamic simulation in presence of some water. So because our body more than 70% made up of water. In the water environment, how it behaves. Whether the docking complex should be stable in the binding pocket or coming out of binding pocket. That can be decided. So this is click the. So this is a DESPON model. So this is the binding pocket. So this is the drug, this is protein. So these are the protein with water molecule. These are the red color is water molecule. This is the protein. This is our ligand. So throughout 100 nanoseconds, it is available in the binding pocket itself. Sometimes it's coming out of binding pocket also. That we have to analyze. So this is a molecular dynamics result. So this is the output for the molecular dynamics. So it is variable throughout the 100 nanoseconds. So all the, this is the ligand, this is the glucose protein, this is ligand. So all together goes in the within the limit. So all are always in the binding pocket. So it is a very good one, more stable. So similarly, RMS value means root means got deviation. So this is RMS value means root means got fluctuation. So where the bond, this is the bond between amino acid and the ligand. So where the bond is there, the fluctuation is less. So here there is no bond, so fluctuation is more. So where the tightly binding bond is there, the fluctuation is less. So based on that, we have to analyze. So these are what are the amino acid will produce interaction. This detailed study. So this is green color is hydrogen bond. So what is this molecule? 346 will produce hydrogen bond with that ligand. So lipophilic bond. These are all lipophilic bond. So similarly, we have to analyze each and every what are the important amino acids are required and what are the amino acids are produced interaction with that ligand with particular bond. So we have to analyze each and everything and how much it is building. You see some it is a continuous band itself. So this molecule is very important. So this is leucine. Leucine 380 is very important. It's continuously connect with that particular binding pocket. So that always connect throughout the 100 nanoseconds, it is connecting. So that are the amino acids are very important amino acids to produce that. Like that, we have to decide. So these are the hydrogen bond. So what is hydrogen bond? These are the pi-pi stack. Tryptophan is a aromatic amino acid. So this amino group, this aromatic group belongs in between two aromatic groups. That is pi-pi stacking. That is important. And this also the violet color is a hydrogen bond. So these are the amino acids are required. And then we have to we don't touch this one. So we have to modify other groups. So these are the different properties we have to do. So RMSD value and molecular surface area, carbon accessible surface area. So we have to analyze each and everything. Then finally conclusion. So what are the compounds? So this will give the first we will see Blake score. The top Blake score against the ERF is minus 12.64 for HCl2 minus 7.842 and for small minus 7.15. When compared to standard drugs, these are the drugs are more active. Like that, we give conclusion. Okay. So this is the advantage of computer-aided drug design. So it comes after 2000 only. Computer-aided drug design. Previously, we are using only that normal. So we'll see how much drastically improved number of publication after 2000. So only we'll do the computational design itself. We have to publish paper. So for generally for one project, it will take 1 year to complete all the process synthesis and publish. Here within 1 week you will completely work out. We'll do all the tests, all the computation method. We have to publish one paper. So drastically after 2000 now up to 5,000 papers per year will come. So this is the main impact of computational drug design method. Because we have to spend very minimum time with maximum investment. So these are the advantage of computational drug design. So thank you very much for listening. So when you're out, ready to clarify.
Gives me immense pleasure to chair this session for Dr. Kalai Rajan Rajgopal. he has drawn the attention of each and everyone of this hall with his wonderful talk. He explained very well about the computer drug design and sir has explained that the advantages of computational drug design over the traditional drug design, like time saving of time and a lot of money, and he explained about the various pages and the steps involved in the computer-aided drug designing and new technologies in the computer-aided drug designing. And he explained about the difference between the ligand-based drug design and structure-based drug design. In the end, he in and out about the molecular talking by taking the animations and case studies as well. And also they provided us the information about the softwares which are useful in this computer type designing. With your I hope with this motivational talk and inspirational talk, some of your students may step into the drug discovery and will become a good scientist in future. Thank you very much for me giving this opportunity in this mega again. Thank you, Dr. Vidyadan. Thank you very much.
Thank you, sir. Thank you, organizers, organizing secretary, management, and Mr. Vikram Chowdhury, Operant Pharmaceutical Federation. Thanks everyone for giving this opportunity. So if you have any doubt, then contact future. So we are doing this as a consultancy and collaborative work also. If you want to learn, you can come there to our college. Or you can send for your project, you can send your samples, so you will do all the work and will give the report on the consultancy basis. Thank you.
Thank you, sir. Now I invite our principal and the organizing secretary to please come on to the dais and do the felicitation to the speaker and also to the chairpersons. First, I request the speaker, Dr. Kali Rajan, to please come forward and accept the felicitation. Now, I request the speaker to please join his hands as we felicitate our chairpersons. First, I request Hanumaiah sir to please come forward. Next, I request Dr. G. Vijay Kumar to please come forward and accept the felicitation. Thank you so much. The oral presentations will start in the same. The oral presentations will start in the same seminar hall. Before we do that, we have an international speaker from Malaysia, Dr. Chowdavaram, our party. We'll be having this oral session for 15 minutes. For 15 to 20 minutes. He's an international speaker and we request him to please come forward and address his session in front of all our audience. And after this session, all the oral presenters are requested to stay back in this conference room for your oral presentations. Now I request Dr. Vasudev our party to please come forward and deliver the lecture.
Sir, give us a minute to set everything up. Please give us a minute to set everything up, sir. We'll start.
Hi, Dr. Nagabhushan. Can you hear me?
Yes, sir. Thank you. Welcome, sir.
Nagabhushanam, can you hear me?
Yes, sir.
Yes, sir.
Yes, sir.
Okay, okay. Are you guys Tamil?
Yeah.
Oh, Tamil alame.
No, sir. Telugu, sir.
Telugu gara. Okay, okay.
Good afternoon. Sir, am I audible to you, sir?
What is that?
Am I audible to you?
Is my my?
Am I audible to you, sir? Is my voice
Yeah. I
clear?
can hear. Yeah. Am I audible? Is my voice is clear?
Yes, sir.
Yes, sir. It's clear.
Okay, that's fine. Because I'm not in my home. I'm in between the travel. I'm in the hotel. So I'm a bit worried. My voice won't be so good. And
Yes, sir.
I will share my, okay. Once you're ready, please let me know. I can share my slide. I can start to talk.
Good afternoon and welcome to the plenary session 6. For this session, we have chairpersons Dr. Suresh V. Chennupati, who is currently working as professor and principal at Chennupati Indo-American School of Pharmacy, Jonalgadda, AP. Sir did his B Pharmacy from Gulbarga University and M Pharmacy in Pharmaceutical Chemistry from Rajiv Gandhi University of Health Sciences. He earned his PhD from Acharya Nagarjuna University and an honorary DSc from Mount Elbert University, United States of America. Dr. Chennupati is recognized for his significant contributions to pharmaceutical education and research. He contributed to the pharmaceutical education and have been recognized for several prestigious awards, including the Bharat Shiksha Ratna Award in 2013 and the APP Young Performer Award in 2014. He has also received Best Pharmacy Teacher Award in 2022 from the Telangana State Pharmacy Council. With this very brief introduction, I welcome Suresh Chennupati to this plenary session. Next, we have the second chairperson, Dr. M.V. Nagabhushanam, who is currently working as principal, professor, and HOD of Hindu College of Pharmacy. He has a teaching experience of 25 years and research experience of 13 years. He has received Best Principal Award 2024 by the Council for Skills and Competencies Government of India. He did his B Pharmacy from Birla Institute of Technology and Sciences, Pilani, Rajasthan, and M Pharmacy from the same institute. And Sir received PhD from Acharya Nagarjuna University in the year 2026. With this very brief introduction, I welcome Dr. Nagabhushanam sir to this plenary session. I request sir to introduce our speaker, Dr. Rajesh Ramaswamy.
Good afternoon to all the delegates and all the participants. And on behalf of Chebrolu Hanumaiah Institute of Pharmaceutical Sciences, it is my privilege to be a part of this wonderful session. And I welcome Rajesh Ramaswamy Garu to deliver his lecture, plenary session on current research trends in stem cells, drug discovery and AI. Sir is offering his services in the Department of Pathology as Faculty of Medicine and Health Sciences in University Putra, Malaysia. I welcome you, sir, to this session. It is my privilege to speak a few lines about you, and on behalf of the Chebrolu Hanumaiah Institute of Pharmaceutical Sciences, we request to start the session, sir. Welcome, sir.
Thank you so much, Dr. Nagabhushan. Can I start now?
Yes, sir. Please.
Okay, thank you so much. Let me to share my slides. The slides are okay. Okay, so I can let me to just to. Okay, thank you so much. And before I start my talk, let me to recite few Guru Chan, Guru Brahma, Guru Vishnu, Guru Devo, Maheswara, Guru Sakshat, Parabrahma, Tasmai Sri Guruve Namah. It's always my pleasure because when I'm talking in a conference in India, I can recite a Guru Mantra. So I'm very privileged for that. And today I'll be talking on the current research trends in stem cell drug delivery and also drug discovery. And first of all, let me to introduce the topic to the participants. I will be introduce what is a regenerative medicine that include a stem cell, and then I move to that portion where how the stem cell can contribute to the drug discovery and disease model. And I will talk a little bit on the AI and drug discovery and stem cell. And first of all, it is my pleasure to introduce what is a stem cell. Okay, what is a regeneration? Regeneration is the body capacity to produce a new cell, tissue, and organs. In our common household, you have seen that you have seen the gecko. Gecko is kind of a lizard, right? Have you seen a lizard when the lizard facing any challenges, being what we call challenged by the predator, it will detach the tail. And after a few days, the same lizard will grow a new tail. So that's we call regeneration. But that property is not available for human, right? So when we lost our hand or a leg, we won't grow back. So the regenerative capacity are actually reducing from that very primitive living to the highly evolved living. So in the case of the human, we have the regenerative capacity within our certain organs like a liver, and we also have the wound healing capacity, which is an indication of the regeneration. And you may want to think what makes a regeneration really happen? What is the tool for regeneration? The tool for regeneration is actually stem cell. So what is stem cell? So everybody in this world, we all have the stem cell. A stem cell is a unique cell within the body, within the physiology of the body, within the cell and the tissue of the body that capable to reproduce a cell that been vanished due to the metabolisms and catabolism. So stem cell, they can be a tissue origin, or they could be from the early phase of the life, and they're able to differentiate into multiple cell type. Before I start my talk on stem cell and drug discovery, and I need to explain what is stem cell, then only you able to understand how to use stem cell in the drug discovery. And now, there's a simple introduction on stem cell. Stem cell means it has two character. One, they able to cell renew and the differentiate. If a stem cell, remember, human being will have very limited number of stem cell. So the stem cell has to be self-replicate without losing its capability to multi-differentiate. So they have to cell renew and keep certain pool of the stem cell for the future use. And same time, they also able to produce multiple type of the stem cell. For instance, so we have the blood, right? Within the blood, we have a white cell, red cell, and also the platelets. So all these three cells are came from one single cell called hematopoietic stem cell. In that means, the hematopoietic stem cell must able to keep some constant pool of stem cell for future use, and they also able to differentiate into a white cell, red cell, and platelet. And stem cell can be retrieved from many resources. It could be come from embryo, could be come from fetal tissue, also come from perinatal tissue, such as umbilical cord blood and water jelly of the umbilical cord, and also it can come from any adult tissue. Let's say if I receive a sample from an individual, let's say and a uterus from the lady who has removed the uterus, I'm able to produce a stem cell that belongs to uterus in my lab. So it shows that every organ in our body, it has a stem cell that can be repopulated upon in vitro culture. And the most important stem cell, which is being known at this time, is a pluripotent stem cell. And the adult stem cell, either they can be natural, it means they came from an organ, or we can take any normal cell and we can induce them into a stem cell. So later I will touch on the part induced stem cell. So there are many adult stem cell, which is in your bone marrow, in your dental pulp, in the hair follicle. Do you ever remember why hair is keep on growing? Because the hair has the hair follicle stem cell, right? So every part of the body has the stem cell, and most importantly, for the pharmaceutical use and also for research, we have a special category of stem cell which called inducible pluripotent stem cell. I'll tell you what. In 2006, the first paper came out that has actually it has evolved that the medical field on stem cell. So the Shinya Yamanaka, a Japanese scientist from Kyoto Prefectural University, he has found that taking a normal cell, taking a skin cell, a fibroblast, he can turn the normal cell into a stem cell. This stem cell have a very huge capacity into differentiating into a multiple cell type. So basically what we know, what is our conventional knowledge on stem cell is a stem cell become an mature cell. And mature cell cannot be a stem cell. There is only one-way communication or one-way travel. But Shinmaya here shows that by taking a mature cell, you can revert into a stem cell. Remember, every single cell in our body actually came from a stem cell. So he has proved that by inserting certain genes, he can recapitulate the ability of the mature cell into a stem cell. That's what he has shown here. So today our talk is mainly revolved, okay, on the inducible pluripotent stem cell. Inducible pluripotent cell can be produced from any of your tissue. If you given me your hair follicle tissue or any fibroblast, epithelial cell from that mouth cavity, we can transform them into a inducible pluripotent stem cell. So we are advocating on inducible pluripotent stem cells. So what is a useful? Remember, many diseases nowadays are degenerative diseases. I'm just giving you an example. um, let's say someone having an diabetic. It's just a condition. Someone is having diabetes and he's a patient, and we can take any of his is mature cell, a mature cell from the buccal cavity, and with the adding a factor, we can transform them into a stem cell, inducible pluripotent stem cell. From inducible pluripotent stem cell, we can generate the beta cell that are responsible for the insulin production. Although this is still under the research, but you can imagine the trajectory of the inducible pluripotent stem cell. Inducible pluripotent stem cell is mainly under research, but it has opened many ways into a regenerative research, especially when it's come to a personalized medicine. Okay, and then people have used iPSC, which is the inducible pluripotent stem cell in a research. However, remember, when we perform any laboratory work, okay, we take a cell, we do a 2D cell culture, and let's say I wanted to examine an extract from that plant, medicinal plant. So we will add the medicinal plant extract, either a crude extract or semi-purified, or even the phytochemical, or even the active ingredients or bioactive molecule on the cell type, and we will get the in vitro data. But however, remember, the in vitro data we have under 2D is insufficient because we are not growing the cell under the in vivo condition. So how this could be actually addressed when if we can produce a system that closely mimic in vivo system, animal model, without jeopardizing other factor. This is what we call organoids. Organoids actually came from a stem cell. They're able to differentiate and organize into a cluster. They are more like a ball, right? But they are more like a ball. It means they're able to differentiate into a particular organ. They form a sphaera, a ball. This ball contains almost every cell of the particular organs, and they are behaving, serving as a miniature organs, right? So now instead of the cell, we have an organoids. Okay, that's why we don't call them as organs. Organs which has everything. But organoids is a representative, a reflective of the organs, the capable of reflecting the function, the physiology, the pathology of the patient, of the individual. So by using a laboratory 2D cell culture from cell line or primary cell, which has a lot of limitation, now we can progress to transform our research into a organoid-based research that give you a better result in terms of physiology and the vertical pathology. Why I'm saying this? It's very simple. Like in the case of screening of that phytochemical or screening of the certain what you call environment pollutant, not all of them work similar on the human and the animal. Okay, whenever we do our toxicity, whenever we want to do some efficacy work, we use animal model because you need an organism. However, not necessarily the animal model reflect the physiology, a pathology of that human. For example, like a methyl mercury is one of the pollutants, one of the toxicants. Okay, when we use this toxicant, the methyl mercury, into an animal model, a cell that derived from animal stem cell, they don't give so much of the impact because their physiology different. But this cell, when being tested on organoids which came from human, it give you a wonderful result. It shows that how much the methyl mercury, which is responsible for Minamata disease in Japan, effect. So sometimes the animal model can fail us because the animal cell and also the human cell, they may vary in terms of the sensitivity or responsible or how they respond to the chemical or phytochemical or any pollutant. That's why nowadays the science are more opt for human derived or a patient-derived cell that has an organ structure that able to function in terms of physiology and pathology. So now you have the idea why organoid is very important, because it gives you an accurate, a close resemblance to the human physiology, human pathology, and also clinical significance. Okay, so how to produce organoids? It's very simple, like how we grow cells into tissue culture flux. That's referred as 2D. Same time, we can grow the organoids on that matrigel or the microfluidic in 3D condition. So with that, we are providing the cell, the organoids, and a close resemblance of the human system. And the organoids have been used very drastically. I would say this: if you type organoids and the drug discovery in the PubMed, you will see many articles that are actually utilizing organoids to do disease modeling, drug discovery, and toxicity screening. Even they are actually using this for the drug repurposing. I'm going to I'll be briefly touch all of them in my talk. So now, so we know that organoids are an important and a very attractive tool in the in vitro laboratory work. It can be used in a many aspects. It can be used in disease modeling, because why disease modeling is very important. Let's say someone having a genetic disease. This is a very rare genetic disease. If I use any other cell from the outside third party, so what happen is not necessarily the result we got are actually reflect of the patient. Now we have the option, so we can take the cell from the patient, or we can take a normal cell from the patient, make them a pluripotent stem cell, and from that we can form the organoids. Now I can personalize, customize testing the drug that going to be work on the patient, because the cell will have a similar physiology, a pathology that belongs to the patient, not the third party, not the animal cell. So that's how we can model the disease. The other one is also related to personalized medicine. So we can do the drug sensitivity. Let's say for cancer, not necessarily all the patient will respond or sensitive to a particular drug, because cancer itself heterogeneous. Not everyone will have same type of the cancer. So we can take the patient's cancer cell or patient cell, and we can make an organoid, and we can test the drug sensitivity, and also we can test the drug adversity as well. The adverse effect and also the therapeutical effect of the drugs can be captured, okay, can be gauged through the organoids. But here I'll be more focused on drug discovery. So here, this is just a schematic figure, what we can do with the stem cell and also the pharmacology of pharmacy, right? So we have the stem cell, and we can use the phytochemical, we can use the drug, we can use any crude extract that enhance the growth of the stem cell. As you know, that stem cell is not an easy cell to grow, and most of the time we cannot use stem cell for patient application because there is a lag in expanding the stem cell. Can we remove the lag period of the expansion by use any phytochemical? I will show you later the result. Or we can also use stem cell to deliver the drug. And the advanced phase of the stem cell is through the organoid. So what we can do, So what we can do is we can produce the organoids, and we can test our drug lead or any phytochemical, the compound that you wanted to test and you wanted to bring at the clinical trials. Okay, that can be used for drug discovery and disease modeling and toxicity and adverse activity screening and drug repurposing. So I will be more focused on drug discovery and also toxicity screening tests here. Okay, this is how phytochemicals are actually, because we have to see what is the impact of stem cell on drug discovery. The one part I will come later. Before to go to the later part, let me to tell how could a phytochemical drug can enhance the quality of the stem cell. As you know that stem cell, when we say a phytochemical, there are so many phytochemicals from that mother nature, from the herbs, especially India, a land with that ample of the herbs, ample of the rejuvenating herbs, like in a Siddha medicines and also Ayurvedic. So we have a Gaya Kalp. Gaya Kalp is actually regeneration. So before anyone have coiled the word regeneration, our Indian scientists, our Siddha, the sages, they have recoiled the word Gayakal. And also they prescribe certain herbs that able to contribute to Gayakal. So like Amla and also Terminalia and also Cassia. There are a lot of the herbs in India, also other part of Asia that able to contribute to the growth of the stem cell. Although the modern research are not so much on that crude extract, but the phytochemical isolated from these herbs are actually has a benefit effect on growing stem cell. And one of my work is I work with the Moringa olifera. Moringa olifera is we call as Murunga in Tamil. I'm not very sure how we call it Telugu. So what I have done is I have extracted the Moringa olifera, and also we have consumed the phytochemical isolated from the Moringa olifera. So when we co-culture the stem cell with the Moringa olifera extract, even the phytochemical, they able to increase the growth of the stem cell. So instead of having a few millions of the stem cell, with the Moringa olifera, we able to capture a multiple fold of the millions of the stem cell. Remember, the number of stem cell is important when we are treating a patient, when we are using the clinical trials. Let's say a patient is needed a stem cell within 2 weeks time. If their stem cell are not growing, we can use the phytochemical to speed up the expansion rate of the stem cell. So what I have shown here is it increased the number of stem cell and also the mechanism of the proliferation. So what we have seen is when we use Moringa olifera extract, it induced the stem cell into cell cycle. Cell cycle is an important tool, cell to divide. So it push the cell into a cell cycle. Same time, it also reduce the cell death, the apoptosis of the stem cell, because cells are growing in the in vitro, an external environment. When a physiological cell are placed in external environments, they are very susceptible for their cell death. So by using the phytochemical, not only we are increasing the growth of the stem cell, but we also reducing the automated or the programmed cell death, which call as apoptosis within the stem cell. So for these experiments, we have used the mesenchymal stem cell, and we also have used the crude and ethanolic extract and the niazurine and niazuridines, the phytochemical that been isolated from that Moringa olifera. Now, so that's how stem cell can be supported with that phytochemical. Now, how we can manipulate stem cell to treat diseases like any drugs or phytochemical, they have their own bioavailability due to physiological system, due to metabolism in the liver, ADME, right? As everyone is aware, they are unable to reach the site of the disease. For example, for tumor, for cancer, right? The cancer cell able to prevent themselves from attacked by the anti-cancer drug. So what people have done recently is, instead of giving a drug or the compound that has the anti-cancer effect, but they're unable to reach the cancer, they have integrated the medicines, the drug, the compounds into a stem cell. One of the stem cell that been utilized to do this, a mesenchymal stem cell, because mesenchymal stem cell naturally they able to migrates towards the tumor. So when a tumor patient, okay, we can combine the drug within the mesenchymal stem cell, and mesenchymal stem cell will bring this drug without undergoing the liver metabolism, and they will straight away bring into the tumor. So with that, you are actually increasing the bioavailability of the cancer drug at the vicinity of the tumor. So that's how people have used a stem cell to deliver the drug. For example, this is one of the example is for glioma. Glioma is a cancer in that cancer in the brain. So people have used interleukin producing MSC. So the MSC are genetically modified, so it produces interleukin 2, and the glioma cell are sensitive towards the interleukin 2, and the MSC, the stem cell, are naturally migrating towards the tumor cell. So by injecting this stem cell, which producing interleukin 2, they able to reach the part where they can see the glioma, secrete the interleukin 2, and eradicate the tumor. Right? So such a therapy is available. This is how stem cell can be used as the drug delivery vehicle. And now, how we can use a stem cell in the drug discovery? Okay, this mainly through iPSC. As we know that the stem cell, okay, the stem cell are being used to treat the diseases. Now, how stem cell can be used to be useful in the drug discovery? For this, the major important stem cells are inducible pluripotent stem cell. Why I say inducible pluripotent stem cell is very important? Because they're able to differentiate any part of the body. You may ask me, why we can't use any other conventionally available stem cell, like a mesenchymal stem cell and hematopoietic stem cell? We can use, but remember, the differentiation potential of the adult stem cell, such as hematopoietic stem cell and also mesenchymal stem cell, is very limited. Like a mesenchymal stem cell, they only can differentiate into a mesodermal, like adipocyte, bone, cartilage, fat. The hematopoietic stem cell, they're able to differentiate into a white cell or red cell. Okay, so however, let's say a patient having a kidney disease, let's say a patient having a pancreas cancer, pancreas diseases, right? So it's impossible to retrieve the stem cell from pancreas. However, we can use the patient single cell and transform them into iPSC. From iPSC, we can produce an organoids that came from pancreatic lineage. So now I can use the patient organoid that came from pancreatic lineage to test, okay, to do the disease modeling and also to test specifically which drug will work for this patient personally. So this will avoid the genetic disparity between two individuals. If we use any animal study, okay, although the system, the genes are almost same, but not necessarily the animal model will reflect the disease, right? And in a drug discovery process, we have four components: the disease modeling, target identification, compound screening, and toxicity testing. Within these four pillars, the three pillars can be enhanced, supported by the stem cell, except for the target identification. Disease modeling can be enhanced by the stem cell. Compound screening, when we can produce a specific cell, not necessarily you will have the cell for Pacific system. But now with the iPSC, we can differentiate into a Pacific system. So we can test your compound on the Pacific system, and same time, we also able to do a toxicity screening. Now, disease modeling is very simple, and what people have, why we need to use organoids or stem cell for disease modeling? The currently conventionally available models such as animal models, single cell, 2D culture, yeast, or any immortalized cell line, they have their all limitation. Like although the cell line, right? Cell lines are a cell that been activated with the oncogenes. They continuously grow, and they most probably not reflect the actual gene constitution of the patient, right? So that is the differences. In terms of animal cell and human cell, what happened is not necessarily the animal cell reflect the physiology and pathology of human. In the case of that methyl mercury, which is cannot be captured through the mouse cell. So the animal model as well do not fully approximate the human physiology. So even the cell line, even the 2D laboratory in vitro culture or the in vivo model, such as an animal model, they are not in a perfect system that reflect the human disease. But we have to use this. You can't use that safety testing. You can't use that proof of concept on the human being. You can't. You only can do this when the drug comes to clinical trial. And before that, you have the in vitro works and you have that preclinical model. And these two may fail the researchers because they behave differently. That's why we need a human base that closely mimic. So in a human base, we cannot use a human to test that toxicity. I mean, to test the efficacy, how it's worked. That's why we came to a stage that we can use organoids that able to reflect the in vitro model and also the cell specificity that particular to a human and also particular to a disease model, and more into very extent into a personalized medicine. For example, the one of the few companies in Japan, they have well developed, well extend into what you call the drug modeling, and one of them is in Ncardia. Ncardia, they produce iPSC, and they use iPSC and also iPSC derived organoid to test all the physiology and pathology that related to the cardia cell. So instead of testing this on the animal model, you can test them in the 3D model of the organoids, the different type of the electrophysiology, the biochemistry, and also the contraction assay that can be done very clearly in 3D system of the organoids. The other one is toxicity screening and stem cell and AI. This is what you have seen is Dr. Son from the Japan. So what they have done is, because we are coming up with the plenty of that what you call the new compounds, and you have no idea what is the impact of the compounds on the human cell in the short period and the longer run. So what Dr. Sean have shown is she has used organoids and she also used the AI. Okay, she combined the organoids, that's what you call that derive from the stem cell, and she has interacted that the organoids with the newly produced compound before release into industry. They have checked the interaction. What is the impact of the compounds on that organoids, and the data which they collected are being what you call channeled to the AI, and AI able to synthesize, able to tell whether the compounds which interacting with the organoids came from stem cell are going to be safe or not safe, whether it has any adverse effect, the adverse effects on the skin or on the any other organs that can be tested on the various type of the organoids. So this has given a new way, because if you use a conventional way, it take a longer time and it will take a plenty of efforts and energy and also the resources. So by introducing, okay, we can shortcut the longest process into the what you call as shortest, and which is going to be a benefit to the industry. And this is a kind of that repeating this this slide. This is a summary slide, how stem cell can be used in the industry, the pharma, and is through a four-way. And nowadays, just remember, people can use straight away directly the stem cell into all these four category. However, the much better version, a stem cell that close to in vivo system, is through organoids. With that, we can do a lot of works on drug discovery and also disease modeling, toxicity, and drug repurposing. Drug repurposing, as you know, that you can use an established drug for different indication, right? That also can be used the organoid. So with this, so I end my talk. I would like to thanks to the Madam Chairperson and also Dr. Prof. Suresh and Dr. Naga Bhushanam for the opportunity. Thank you so much. If there is any question, I'm much happy to answer.
Thank you very much, sir, Professor Rajesh Ramaswamy garu, for your wonderful session. Sir, from my end, I request for one of my doubts to clarify, sir. Am I audible, sir?
Yeah, you're audible.
Right. Sir, about the usually for conducting these clinical trials, we take the permission from the regulatory authority in the form of clinical trials for phase 1, phase 2, phase 3, something like that. So,
Definitely.
how about these utilization of the stem cells instead of or in place of the animal models? The regulatory clearances for getting permissions. Is it yeah.
Thank you so much for the question. It's based on that regulatory system you have in your country. At the moments when the FDA or any other European regulatory body, when they assess the efficacy of the safety of the drug, at the moments you can't replace the animal model, right? However, they also recently they also have um, they have removed for certain cases the requirements of the animal model because the animal model is not relevant. So in that case, they will request for any model that equivalent to the animal model. In that case, the organoids can be a very good choice for the testing. Why I'm saying this? Because although organoids is not fully being utilized or to be approved by the authority, however, the process are still going on. As a scientist, we should provide valid data to a regulatory body or to authority to show that the result is valid. And as you say that, this is still a research. That's why I say current research trend. It is not something that been established. No, organoids are still new. It's only in 2006 people have discovered the organoids, and the research in organoids is still very, I would say, is not so much of advanced. So what we can do is, if let's say for some diseases you can't have the animal model, right? Diseases that are related to their brains and also the psychology, or for any rare genetic diseases where the animal model is not available. In that case, we can utilize a specific model that can be derived from the organoids, right? We can test them. So although it has potential to replace the animal model, I say potential, but at this moment that is not a possible. So because we have to follow the existing protocol. If your country require or instead, okay, or they insist on that animal model, you still have to do this. However, a result which come from organoids does give you a tremendous research data, and sometimes it may give you result which is a very clinically significant. So at the moment, the use of the organoids are very limited, not so much into that what you call the pathways to the drug the how do I say drug discovery.
Thank you, sir. Thank you very much. Thank you very much, sir, for topic being covered very, very in-depth illustrations that were given with respect to the stem cell characterization and starting from its sources, and also giving an indication about what is recapitulation and the inducible pluripotent stem cells. And most of the diseases are regenerative diseases. This is one of the excellent statement what you have given, and the role of these stem cells, especially at the capacity as a regenerative cell, and also about the types of various roles that the organoids are playing with respect to disease modeling and drug discovery, toxicity, and at the levels of the personalized medicine and regenerative medicine, the role of the organoids are beautifully explained by you, sir, and the use of the stem cells and their derivatives at both the experiments of the pharmacology and also in the role of the pharmaceutics, and how these phytochemicals are also enhancing the quality of the stem cells. Especially you have talked about the moringa leaves and the Moringa oleifera, the promotion of the proliferation and reduction and absorption of the stem cells. So all these things were beautifully explained by you, sir. And stem cells as the drug delivery vehicles is one of the most useful concept, and the drug discovery and the disease modeling, how these stem cells are playing their vital role, and also in the toxicity screening, these stem cells are playing vital role. So we are very much thankful for your wonderful session and your insights in the selection of in the selecting this current research trends in the stem cells drug discovery and the role of AI in the process of drug discovery, and especially the replacement of these organoids in place of these animal models will definitely reduce the burden of the clinical trials and also on the experimental animals, what I feel from this session. And we are very much thankful for your time and the insights about the current research trends in stem cells drug discovery AI, sir.
Thank you so much, and my great appreciation to you, sir, and also Madam Chairperson and Prof. Suresh, and also Dr. Vikram. Thank you so much.
Thank you, sir.
Thank you.
A very good evening to you all. For the last time, I welcome you all to this valedictory event of the two-day international conference organized by Chebrolu Hanumaiah Institute of Pharmaceutical Sciences in association with Operant Pharmacy Federation and also Indian Pharmaceutical Association. The title of the two-day international conference is The Role of Artificial Intelligence in Drug Discovery and Drug Development. In these 2 days, we had multiple plenary sessions, multiple e-poster sessions and oral sessions. And today, I stand in front of you as we successfully completed the 2 days and we are now about to conclude the conference with a valedictory event. Without any delay, I would like to invite all the guests on the dais so that we can start the valedictory. First, I invite on stage the organizing secretary of the entire conference, the person who took care of the entire 2 days, followed by multiple days before the conference, Dr. V. Venkatrao, onto the dais. Next, I would like to invite on stage the person who directed him all throughout, Dr. S. Vidyadhara, the principal of our institute. Next, I would like to invite on stage the man behind Operant Pharmacy Federation, the one who we have been associated since a couple of years, Dr. Vikram Chowdhary, onto the dais. Next, I would like to invite a group of guests who are here with us to share this wonderful valedictory event. At the outset, I would like to call on stage Dr. M. Prasad Rao, Principal, MAM College of Pharmacy. Next, I would like to invite on stage Dr. K. Venkatraman, Principal, ASL College of Pharmacy. Next, I would like to invite on stage Dr. Y. Ankamat Chaudhary, Principal, NRA College of Pharmacy. Next, I would like to invite on stage Dr. Narendra, Principal, BJS College of Pharmacy. Next, I would like to invite on stage Dr. A. Vasudev from Malaysia, International Malaysia University, onto the dais. Next, I would like to invite on stage CH Balashekar Reddy Garu onto the dais. Lotus Laboratories onto the dais. Next, I would like to invite on stage Dr. R.S.C. Sasidhar, Professor, Chebrolu, Hanumaiah Institute of Pharmaceutical Sciences. Next, I would like to invite Dr. A. Ramu, Professor, Chebrolu, Hanumaiah Institute of Pharmaceutical. Next, I would like to invite on stage Dr. K. Subharao, Dr. Thank you all. Now I request all the guests to please be seated. We'll start the proceedings with a prayer song.
Sadaya Giriraj Sudha Tanaya Sadaya Giriraj Sudha Tanaya Surana Chita Yuga Surana Chita Yuga Paripalayama Ivarajanuga Paripalayama Ivarajanuga Giriraj Sudha Tanaya Sadaya Giriraj Sudha Tanaya Gananadak Parasansara Gananadak Parasansara Shamavari Nidhi Rajani Kara Gamavari Nidhi Rajani Kara Surana Surana Ranashyam Bhavasri Tyagarajanuga Ranashyam Bhavasri Tyagarajanuga Giriraj Sudha Tanaya Sadaya Giriraj Sudha Tanaya.
To begin this valedictory event, I would like to call upon Dr. Vikram Chowdhary, Operant Pharmacy Federation, who is establishing with this institute for the second time in bringing out this today conference. So I cordially welcome him to please come forward and share his experiences and also to speak a few words about this conference.
Good evening to one and all present here, dignitaries on the dais, all the principals from the various colleges, faculties, and dear students and delegates from across the country who is joining virtually as well as offline. The today's PHARMANEST 5.E International Conference on Accelerating Drug Discovery and Development using Artificial Intelligence and Machine Learning, organized by Chebrolu Hanumaiah Institute of Pharmaceutical Sciences in collaboration with Operant Pharmacy Federation and Indian Pharmaceutical Association AP State Branch. The objective behind this 2 days conference to having the latest technologies and the advancement in the drug discovery technologies. So we had total 8 plenary sessions, 3 in the online mode and 5 in the offline mode. We started from the Dr. Srini John Matthew. She is the research associate at the University of Calgary, Canada. She elaborated about the clinical research and role of artificial intelligence in the drug discovery and design. Next session we had by Dr. Srinivas Reddy, who is the assistant professor at IICT Hyderabad, who elaborated about the mRNA vaccine technologies, how the role of artificial intelligence in the vaccine discovery and the biopharmaceuticals play a key advantage. The next session we had by the Dr. Rajneesh Kumar, who is the associate professor at IIT BHU, and he elaborated about the role of artificial intelligence and the machine learning in the small molecule entities research by a practical examples. I hope this session is very helpful for you. Then in the first day we had the e-poster presentations in the different streams, almost 430 e-posters and the oral presentations were evaluated. And Second day, we had the first session by Dr. Dharmendra Ahuja, Principal Mentor, Bureau of Health Sciences at Government of India, who is given the practical examples of the ex-physio and ex-pharm software, how the pharmacological experiments can be do using the artificial intelligence, the machine learning, how it can be save a life of the animals. He has given the practical examples, and he is the principal mentor present here, who is the man behind having the softwares for the pharmacological studies. So we can survive the lot of the animals using the AI softwares. The second session of the day 2 was delivered by Dr. R. Kalijajan, Associate Professor at JSS College of Pharmacy, who elaborated about the computational approaches for the long libraries, which can having the short duration of the molecule discovery. He given the real life examples of the case studies, how they are doing the good research at the JSS College of Pharmacy, and how other people can associate. They have the collaboration and collaborative research on the AI, how can we perform. Next session is delivered by Dr. Vasudeva. He is the Associate Professor at International Medical University, Kuala Lumpur, Malaysia. He has given an example of role of AI, how a bachelor student can have the AI in the student life role, and what will be the benefits and what are the harms for the students. If you using the artificial intelligence, so you should use your mind behind it. Just yesterday, one statement is got from the one of the billionaires from the India, Mukesh Ambani. He said, everyone we are using the chat GPT. Using for the anything you want, just you typing of the chat GPT and using like what is this. But the using of chat GPT also require your mind. If you not use your mind behind using the chat GPT, what are the knowledge you have, it will be going what? The chat GPT is not giving you the knowledge expansion. It is decreasing your knowledge because you are going to be dependent on the chat GPT. You are not learning anything. If any statement is coming in front of you, you are not aware, you simply do on the search on the Google, or you will be searching on the chat GPT. But in the real life, you not having the any general knowledge. So this general knowledge is important for us. Yes, technologies are coming, artificial intelligence, machine learning, and different technologies are coming. But all the different technologies are the for the right usage. If you use the right technology on the right time, so it will be useful. If you use the technology at each part of the life, then it will be creating a wrong impression on your entire career. So that's why the Reliance industry given a statement, the chat GPT will not a longer term career for the budding professionals because it's not giving a good manpower to the industry. With just span of the 2 years of the chat GPT, the industry facing a lot of the problems because they're not getting the right manpower. Whatever the manpower they are getting, all are the dependent manpower. Anything they want, they just search on the Google. They're not learning the right outputs. That's why we have the such kind of the conferences, workshops, and these things so on. The next session delivered by the Dr. Arup Kumar Mishra, who is the Associate Professor at Department of Pharmacology, AIIMS Mangalgiri, who elaborate the role of artificial intelligence in the pharmacovigilance studies, ADR monitoring, that is very important for the pharmacy students who are joining. The how the ADR and the pharmacovigilance studies can be monitored using the AI tools, and it is very important. Now the pharmacy colleges are reporting the more ADRs as compared to the medical colleges, especially those pharmacy institutions having the medical colleges and hospitals. They giving the more reporting to the Indian pharmacopoeia commission because they are working in the clinical practice. They are working hard and creating a positive impact on the country. The last session was delivered by the Dr. Rajesh Kamaswamy, who is the Associate Professor at University Putra, Malaysia, who had given the role of artificial intelligence in the clinical research and the stem cell therapy. Nowadays, the stem cell therapy is becoming the one of the popular research area in the pharmacology and pharmacy also. So how the stem cells now using the AI and what AI can do in the stem cells, he has given a lot of the practical examples which is helpful for all the budding professionals. Also, we had the oral presentations from the delegates. I hope these 2 days international conference will given you a lot of insights or role of artificial intelligence in the accelerating the drug design and discovery. All the expert speakers meet your expectations. What you had come last 2 days with the knowledge sharing, engagement with the professionals, networking, and the knowledge transfer. I hope all the points you got as per your expectation. I also thank to the management and the principal, Dr. S. Vidyadhara sir, coming forward and having the collaborations with this 5th edition of the PHARMANEST International Conference at this institute, and we are working from last 2 months to having this conference a grand success. I hope this conference is valuable and the knowledge insight for everyone. Thank you very much for joining this 2 days Pharmanests International Conference. On behalf of Operant Pharmacy Federation and entire organizing committee, I thank you very much.
Thank you so much, sir. Now I request Dr. Vasudev to please come forward and give this feedback.
Distinguished members of the various doctors, all participants. Firstly, I would like to congratulate you all for successfully completing 2 days vibrant artificial intelligence based several sections facilitated by eminent speakers and resource persons. Firstly, I would like to congratulate all the members. Coming to the feedback. So feedback is very sensitive matter. So the term feedback, the moment already you all might go on into a negative side of it. Definitely not. Feedback means just for enhancement. We can include both positives as well as negatives. Okay. So my positive feedback, so always let's be positive. My positive feedback, I request all the members to continue such initiatives and organize many events and enhance the digital competencies of the graduate workforce in near future. I hope so. So I would like to request all the eminent personalities here, Chebrolu, Hanumaiah Institute of Pharmaceutical Sciences, Operant Pharmaceutical Federation, Indian Pharmaceutical Association, and distinguished members from APDI. So every member to consider engaging young and dynamic students in such learning activities and networking, create such opportunities to engage into effective decisions and enhance their career opportunities. So I would like to give that positive input from my side. However, always there will be a room for improvement. Of course, this is not negative. Okay, so the room for improvement. We are discussing about AI, accelerating artificial intelligence in drug discovery through AI. So the fundamental concept AI is totally software related. So we are mainly looking into the other side of the coin. So meaning we are looking into the end user pattern. So what is the other side of the AI? So while AI could not exhibit or demonstrate or our expected outcomes, majority of the times we are failing because I have enormous experience since last 12 years using AI software. But most of the times when I come to a prediction and I transfer this prediction to the laboratory experimental condition, totally opposite observations. So why is that so? Because as I mentioned, AI is totally constructed based on data. So the reliability, accuracy of the data what we use to train the AI model is the key component of it. So we should understand that. So simply saying AI is no use. Because AI construction must be based on various scientific, reliable, hypothetical algorithms. So these algorithms must be fully tested, and the tested algorithms must be validated further in the proof of the concept studies and so on. So I think few sections must be from the computer scientists who actually developed AI. That's what my thinking is. What do you think today? What is your opinion on that? So majority of the sections, AI users, not AI developers. Am I right or not? So that's what my input only. So if we bring some eminent speakers who are actually developing AI algorithms, who are actually creating the AI modeling systems, they can explain the background working phenomenon, how the prediction is actually taking place, how the simulation is coming out, so that we will have a very clear picture how AI works. Okay, so that's what my input. So both are complementary. Okay. So one more thing I would like to share some personal experience. I specially come here only for this event because I already utilized my all annual leaves in the last December. I have no more leaves actually. This is totally a professional visit, especially to Chebrolu Hanumaiah Institute from Malaysia. If anyone comes onto the stage, I can show the evidence to you. Total, I need to take 5 approvals. 5 approvals starting from my line manager, HR, so on. So all approvals received, and they encouraged me to immediately participate, go and participate, and enhance young researchers. Okay, do networking with the eminent speakers and so on. They provided enormous support. For that, I would like to heartfully thank IMU University, Malaysia. So, last but not least, you must be wondering why I am specially given that much importance to Chebrolu Hanumaiah. Because my best friend, Dr. B. Professor is now Venkatrao, is my best friend. We were very close. And moreover, if you have very good friends, what is the benefit? I just let you all know. He is the one given me the opportunity to speak in front of public. The first opportunity in the year 2020, I think. 2020, the first opportunity to speak in front of all the participants, online conference, online webinar conference, online conference. So they started, it was a very great start. After that, almost I received 30 or 40 invitations across various places in different states. So I feel like this is like my mother for the initiation of my facilitation of workshops online and delivering speeches, facilitating face-to-face workshops in Bangalore, Kerala, Hyderabad, several places. Almost I participated in 40 sections. All because of the first encouragement and the best encouragement from Chief. Thank you all.
Thank you so much, sir. And we would love to have you here once more, whenever possible. Next, I would like to invite C.H. Palasekar Reddy to please come forward and speak a few words on this occasion.
Good evening to all. Organizers of this artificial intelligence program related to different areas. Organized very well. I hope you got very well. So small request for artificial intelligence experts. So nowadays we are using all users to get some, for example, all you know LinkedIn very well. to get the LinkedIn professional sites, everyone needs to pay around 5,000, 6,000 per month like that. If you have a group of people, activities comes around per year 7 to 8,000 rupees. Like that, any artificial intelligence good network is there. If we buy through our channel, so through our associations, it will be very useful for users like us. So this is my small request. Thank you for giving the opportunity.
Next, I invite Dr. Venkatraman to please speak a few words on this occasion.
Respected dignitaries on the dais, off the dais, my dear students. The today's international conference. This is the topmost priority of the theme of the work. This is new area already. Few speakers are deliberations for artificial intelligence. Already recently, our beloved Chief Minister of Andhra Pradesh, first program, first time in history of India, artificial intelligence to oka program oka Chief Minister conduct cheyadam, Prime Minister kuda cheyale India lo first program Andhra Pradesh Amaravati lo jarigindi. And artificial intelligence importance enti, skills ela develop cheyali students ki ఏ విధంగా చేయాలి ఎలా చేయొచ్చు అనే program లో first Chief Minister గారు ముందుకు రావడం జరిగింది. Already skill development అనేది ఆంధ్రప్రదేశ్ లో all education sector institutions లో already జరుగుతా ఉంది program. So my dear young budding pharmacists and my dear students, మీరు ఎక్కువగా ఇలా కాన్ఫరెన్సెస్ కానీ సెమినార్స్ వర్క్షాప్స్ ప్రతి దానికి అటెండ్ అయినప్పుడు డిఫరెంట్ ప్లాట్ఫామ్స్ మీద ఇండియన్ ఫార్మస్యూటికల్ అసోసియేషన్ అసోసియేషన్ ఆఫ్ ఫార్మస్యూటికల్ టీచర్స్ ఆఫ్ ఇండియా అని ఎనీ ఆర్గనైజేషన్ కండక్ట్ చేసినప్పుడు మీరు మనీ గురించి మాత్రం చూడకండి. అది 1000 రూపాయలు 500 రూపాయలు రిజిస్ట్రేషన్ ఫీజు అని కాదు. ఎందుకంటే డిఫరెంట్ టైప్ ఆఫ్ ద స్పీకర్స్ ని ఒక ప్లాట్ఫామ్ మీద తీసుకురావడం, వెదర్ ఆన్లైన్ మోడ్ ఆర్ ఆఫ్లైన్ మోడ్. మీరు మనం విడిగా వెళ్ళలేము. సపోజ్ మలేషియా అవ్వచ్చు, అమెరికా అవ్వచ్చు, లేకపోతే యూరోప్ అవ్వచ్చు, ఏదైనా కంట్రీకి వెళ్ళాల్సిన అవసరం మనం వెళ్ళలేము. సో అటువంటి కండిషన్ లో ఒక కాలేజీ మేనేజ్మెంట్ ముందుకు వచ్చి ఒక ఆర్గనైజేషన్ లైక్ విక్రమ్ చౌదరి ఓపరెంట్ ఫార్మా అట్లా ఆర్గనైజేషన్ ద్వారా ఇట్లాంటి గొప్ప వ్యక్తుల్ని ఒక ప్లాట్ఫామ్ మీద తీసుకురావడం అనేది చాలా గొప్ప విషయం. సో మీ అందరిని ఒక చోటకి తీసుకొచ్చి ఒక చోట కూర్చోబెట్టి కావాల్సిన విధంగా ఫెసిలిటీస్ ప్రొవైడ్ చేసి వాట్ ఈస్ గోయింగ్ ఆన్ ఇన్ గ్లోబల్లీ, ప్రపంచవ్యాప్తంగా ఏం జరుగుతుంది, మనం ఎక్కడ ఉన్నాము, మనం ఎలా ముందుకు వెళ్ళాలి, మనం ఏం తెలుసుకున్నాము, ఎలా తెలుసుకోవాలి, రాబోయే రోజుల్లో మన స్కిల్స్ ఎలా డెవలప్ చేయాలి, ఏ విధంగా వెళితే మనం ముందుకు వెళ్ళగలుగుతాము, మనకున్న ఆపర్చునిటీస్ ఏంటి, మన ఛాలెంజెస్, ఇవన్నీ కూడా ఒక కామన్ ప్లాట్ఫామ్ తీసుకొచ్చి ఈ టైపు ఇంటర్నేషనల్ కాన్ఫరెన్స్ ఆర్గనైజేషన్ చేసిన నా బ్రదర్ డాక్టర్ ఎస్ విజయాధర్ గారికి, అలాగే మేనేజ్మెంట్ చెబ్రోలు హనుమయ్య ఇన్స్టిట్యూట్ మేనేజ్మెంట్ వారికి, అలాగే వేరియస్ ఆర్గనైజేషన్ అంటే ఈ ప్రోగ్రామ్ కావాల్సిన ఆర్గనైజింగ్ టీమ్స్, ప్రతి టీచర్ గారు, అలాగే ప్రతి విద్యార్థి, ఈ ఆర్గనైజింగ్ గ్రూప్ లో ఉన్న ప్రతి వారికి కూడా పేరున నేను ఆంధ్రప్రదేశ్ ఫార్మస్యూటికల్ టీచర్స్ ఆఫ్ ఇండియా తరఫున యాజ్ ఎ ప్రెసిడెంట్ గా నా ఆఫీస్ బ్యాలర్స్ అలాగే నా టీం తరఫున మరొకసారి అందరికి పేరున ధన్యవాదాలు తెలియజేస్తూ ఈ అవకాశం ఇచ్చిన నా బ్రదర్కి మరొకసారి ధన్యవాదాలు. థ్యాంక్ యూ.
Thank you, sir. Now, I request our principal to please come forward and do the conference report.
Good evening. Firstly, my sincere apologies to all of you for holding you up to 9:30 yesterday without providing dinner. Sometimes when we invited very important guests, it is going to happen. And he has given us the schedule at 4 o'clock and he arrived here at 6:30 p.m. yesterday. And the program went on delayed
just because of several managements who have come here to felicitate the very important guest. Anyway, thank you all for being with us for the last 2 days and making this program, this international conference, a grand success. So, when we planned this 2-day international conference way back in November, so anyway, we are connected with the most learned association, that is Operant Pharma Federation. He suggested to keep this in the month of January last week. The same type of international conference we have conducted last year during November, that is 23 November. So we thought of conducting in the same November, but due to exams for several students under Acharya Nagarjuna University, we kept it postponed till today, and in January we made it. And at the same time, as you all know that on the same 2 days, parallelly here and in another college adjacent to us, one more international conference is going on. However, there is a tremendous response from 46 colleges, registering more than 500 student delegates to this particular conference, with 394 abstracts that are present. And to make use of this particular conference, we have organized this in hybrid mode. Some of the delegates have participated through online mode, and here there is one device to that. It is live streaming to the people who are not able to come to this place. And due to extraordinary response from various colleges and dear students for presenting their papers, we were shocked how we can accommodate all these 394 papers in a span of 2 days, and in a span of hardly the slot that was given is 2 to 4 hours. So then we decided all the e-posters of our college, about 70 or 70, were evaluated on 28th, and the next highest number of abstracts that were received from Vignan Institute of Pharmaceutical Technology, that is Subhada Visakhapatnam. We conducted the online poster presentation process on 29th, and parallelly, all those who could not able to come here, for them we conducted the live presentation through online mode through Zoom platform. So in such a way, we could able to dispatch all the things in a very streamlined manner, and we could able to complete all 394 papers in a span of 2 days, and now we are very much comfortable. And today also, I just wanted to leave you little early because yesterday it got delayed like anything, and at least today we thought of leaving you from this area by 5 o'clock. Now anyway, it is already 5. In a span of now, you are all very much enthusiastic, and being an youngster, once you present a paper, you are eagerly waiting that whether we got prize or not. But I would like to say, your presentation itself is a big prize. So it is not that if I give prizes to all 395 posters, like first, second, third, fourth, fifth, and 394th prize, there is no value. So some about 40 students are going to get prizes now, and this is going to be distributed right now. And remaining all, don't feel that you did not get anything, but you are also respected very well in the form of your abstract is indexed with ISBN number, so that is highly valid, and it is considered everywhere for your academic upliftment as well as in your colleges, NAC and other accreditation processes. And at the same time, all the research activities that you have presented here, and you are now eligible to publish your research article in one of the Malaysian journal, which is indexed by Scopus through this conference. A special issue is going to be released, and if you are able to publish, you can publish it through our platform. That is the another provision made through this conference. So that means all 395 are rewarded and your abstracts are highly indexed with ISBN number. So it is not a simple document that you have simply came here and submitted and presented. It is very valid document, and your certificate is also having a barcode and number that is linked with the DigiLocker. Okay. And when it comes to artificial intelligence, as Dr. Vasudev Rao and their friends and respected experts delivered their talks, no doubt we are end users of artificial intelligence. And no doubt there need to be some expert type people to develop certain kind of tools for utilizing this particular artificial intelligence technology. So that's why very recently also in our college, I think November 29th, we have conducted a pharmathon in association with our sister concerned RVRJC College of Engineering. So they supported us, and there was a cash prize of 20,000, 15,000 and 10,000 for the prototypes that were developed by our friends. And few of our pharmacy friends got prizes, and those prototypes, and you can further submit to our RVRJC SDTP, where we have an incubation fund. Through that, we also support you for commercializing that particular prototype that you are going to develop. So this particular prototype development will be encouraged and supported in future, because we have a lot of fund at our RVRJC College of Engineering under the incubation center. Almost we have kept 5 crores for that particular one. And any student from any college, from our pharmacy fraternity, if you wish to come out with any prototype product, then we will definitely recommend to our RVRJC. Through that, we will support you financially. So there is a provision that from up to 10 lakhs, we can provide you as a support, and thereby you can make that one as a commercialized one. So that is the one I wish to mention. And here, when it comes to utilization of artificial intelligence, there are some kind of indirect, what do you call, some fear in the members of various professionals. If artificial intelligence emerges, there is a huge setback of employment. Don't worry, we are already using artificial intelligence in several areas and in several platforms. But without manpower, without our knowledge, and without our basic information and science, artificial intelligence cannot do anything. You have already seen a cinema called Robo. So if you utilize the Robo by incorporating the proper inputs, it works properly. If you give the negative signs and negative inputs to that, it is going to become a big villain to all of us. Similarly, in this case also, artificial intelligence, if you use it with your knowledge, with your basic science and knowledge, it is going to give you highly potential experimental knowledge, and thereby we can save time, save money, and we can do the wonders in this particular one. And one simple example I would like to mention here, Dharmendra Ahuja sir is here. He is the inventor of pharmacology software, which we are all using in our colleges. when we were studying pharmacology means we have to go to the frog stock, we have to take the live frog, we have to stun that. After when it is paralyzed, that has to be brought onto the dissection tray, it should be dissected, we have to do the experiment and so on and so forth. Now, as per CPCSCA and other regulations, experimentation on animals at UG level is strictly banned at any level. So just because of that, we have to depend on some simulated softwares. When it comes to simulated softwares, just 5 to 6 years back, there were no simulated softwares. What we have done, we have simply given the data, we have simply given the text material to write it in the record and to draw the figures and just submit it. And we must thank Dr. Ahuja sir for developing such a nice simulated software which gives us some simulated experiments which you are all doing it at our pharmacology laboratories. So in our Andhra Pradesh, through JNTU consortium, all our pharmacy institutions formed a consortium. Through that consortium, almost 80 colleges have subscribed this particular X-Pharma software, and most of the pharmacy colleges are utilizing this particular one. And they also have developed a software for communication skills that is also there, and also they developed some plagiarism software also. So here we have a live example who developed several tools which are highly required for us to utilize our artificial intelligence in our health sciences. And when it comes to the other end, what is the problem with artificial intelligence? One simple example I would like to mention, which is my direct experience, is when I was doing my PhD program, when we were conducting in vivo pharmacokinetic studies on animals, we used to inject or administer the drug, take the blood samples, analyze the blood samples. After analyzing, we are going to calculate several pharmacokinetic parameters: rate of absorption, elimination half-life, Pmax, Tmax, half-life, area under curve, maximum residence time, etc. We used to calculate. All those we used to calculate by doing the calculations which are there in your Brahmankar textbook. Okay. Now we are grateful that several softwares are established. So just by giving the data, by giving the input of values that are arised out of the blood samples collected and the OD values or whatever the concentrations that are arrived, just by giving that, we can calculate all these values. But one glitch at this particular stage is biological half-life for any drug is its inherent property. It is not going to change. But in one of the software that is nowadays being used by several researchers, that is USA Summit software, for calculating various pharmacokinetic parameters, the half-life is also going to change. Particularly when it comes to do the in vivo experimentation process on extended release formulations. So in the extended release formulations, we do the release profiles for more than 12 hours and so on and so forth. In that case, of course, maximum residence time can be increased, but half-life should not be changed. That is inherent property. But in this case, the half-life of that particular one is also greatly increasing. That is the only mistake that we are going to face. And even in the docking software also, we are doing the docking process for developing a new drug molecule. Sometimes the ligands that are going to bind to the protein are going to vary with the original experimentation that we are going to get it. So this artificial intelligence is helpful and aided for us to save the time and to minimize the experimental losses during our processes. but it is not going to sack any of our employment or any of your perspectives in any more future. So don't be panic with this artificial intelligence, but make use, being an artificial intelligence user, make use of this, get benefit out of this, save the time, save the economy, and do the better things out of it. With these few words, once again, I am highly thankful to all the colleges and all the dear students who have come from various colleges to be here with us, and hope that you are all comfortable here, enjoy 2 days stay at our college, enjoyed our hospitality, and wish you all the best. Thank you.
Thank you, sir. And as principal rightly said, everybody are waiting for the prize distribution. But before that, we have a small felicitation event. For that, I request all the guests on the dais to please felicitate few important persons. First, I request Dr. Vikram Chaudhary, Operant Pharmacy Federation, to please come forward and receive the felicitation. Next, I request Dr. Venkatramana to please come forward and accept the felicitation. Next, I request Dr. Chowdavaram to please come forward and accept the felicitation. Now I request Dr. P. Vijayata and Dr. P. Sindhu Vaishnavi to please come on to the dais and announce the prizes.
I request Vikram Chaudhary sir to give away the prize. Firstly, pharmaceutics. Consolation prize is shared by two participants. K. Sarthika of Priyadarshini Institute of Pharmaceutical Education and Research. Shreya Sujan Buddha, KVSR Siddhartha College of Pharmaceutical Sciences. Third prize on the Pharmaceutics Department is shared by 3 members. Firstly, N. Diksha Padmaja, Chebrolu Hanumaiah Institute of Pharmaceutical Sciences. Malineri Perumallu Pharmacy College.
M. Jahnavi, Hanumaiah Pharmacy College. Y. Sai Meghana, VJS College of Pharmacy.
First prize goes to Chowdavaram, Research Scholar, Acharya Nagarjuna University.
Second prize goes to Shravan Kumar Avati, BDVS HSK College of Pharmacy. Now I request Dr. Prasad Rao sir to please hand you all the prizes.
In pharmaceutical analysis, consolation prize goes to B. Gautami of Chebrolu Hanumaiah Institute of Pharmaceutical Sciences.
Third prize goes to V. Pravalika, VJS College of Pharmacy.
Second prize is shared by two participants, P. Kumari of Chebrolu Hanumaiah Institute of Pharmaceutical Sciences and Dr. G. Patna, Andhra College of Pharmacy.
First prize goes to P.H. Jaswant Kumar, Research Scholar, Acharya Nagarjuna University.
Thank you very much, sir. Now I request Venkatramana to give her the prizes. In pharmaceutical chemistry, third prize goes to A. Venkata Harshita of MAM College of Pharmacy. Third prize is shared by three participants, of which Venkata Harshita is one. Second prize, third prize. Dr. K. Nagalakshmi of MAM College of Pharmacy. And M. Tulasi Ramalakshmi from Malini Permano Pharmacy College.
Second prize goes to Kanna Pavani, ASL Pharmacy College.
And of University College of Pharmaceutical Sciences, Acharya Nagarjuna University. And Tulasi Ramalakshmi, Malini Permano Pharmacy College. Second prize is shared by two participants, Kanna Pavni of ASL Pharmacy College and T. Ravika. T. Maunika of University College of Pharmaceutical Sciences, Acharya Nagarjuna University.
First prize goes to T. Bhuvana of Chebrolu, Hanumaiah.
Thank you, Venkatram sir. Now I request Chowdavaram sir to give away the prizes in pharmacology department. Third prize is shared by three participants, T.H. Ratna of Hindu College of Pharmacy.
I. Karishma of NRI College of Pharmacy. Yes, Sujita of MAM College of Pharmacy. Second prize goes to J. Samuel of A.M. Reddy Memorial Hospital. First prize goes to Sheikh Iqbal Nas.
Thank you, Andhra sir. Now I request Santama Chaudhary sir to give away the prizes. In pharmacy department, third prize is shared by two participants, M. Lavanya of Queen's College of Pharmacy and M. Kiran Kumar of Chebrolu Hanumaiah Institute of Pharmaceutical Sciences.
Second prize goes to K. Nizamuddin.
Victoria College of Pharmacy, second prize. First prize goes to Ramya Reddy of Chebrolu Thank you, Ankama Chaudhary sir. Now I request Park Secretary sir to give away the prizes. In clinical pharmacy and pharmacoeconomics, consolation prize goes to Sheikh Reshma of Chebrolu Hanumaiah Institute of Pharmaceutical Sciences.
Third prize goes to Vice Secretary of Chebrolu Hanumaiah Institute of Pharmaceutical. Campus of DVSS HSK College.
She having a Chandramoulipuram in the school of pharmacy. By having second one goes to the Ministry of Trade Health and Economic Pharmaceutical Sciences.
Now coming to the oral presentations. The second prize goes to Dr. Lakshman Vijapur.
Second one is also chaired by Dr. C.H. B. Pravina Devi of Joginapalli PR Pharmacy College.
For online oral presentation.
And the first prize is also shared by 3 participants, of which B. Swati from VJ College of Pharmacy.
Dr. Sachin Kaur, Chavdavaram College of Pharmacy.
Dr. K. Vasudeva from International Malaysia University, Malaysia. This is type of prize distribution. I request Dr. Raghav Patel to propose roll up this.
With someone vote of thanks. For the last 1 week, our staff and our students have tirelessly working for the conduction of this 2-day international conference, and they made it real and made it magnanimous and more colorful. And let us all give them a big hand of appreciation. I request all the dignitaries to please be seated. A disclaimer to the audience. A small disclaimer to the audience. Vote of thanks will last for 15 to 20 minutes. My God. Okay, good day, Vinandi. My God, Parinsanti, Kani Pulswati. First of all, I'm very, very happy that the stage is full of
people from educational background, because I think if there is anybody who understands the effort that is behind a conference at a business, so only those people know the importance of the difficulties that one could face while at conference. And I ask their permission, please give me 15 minutes so that I let everybody know what are the efforts that we underwent in the last 10-15 days so that we can make it possible. So please give me 15 minutes. Thank you. Thank you. I'll make it interesting for you. I hope I'll not make it bored, but I'll try to make it interesting. Because I think it is important for all of you to know what were the efforts that you were not able to see. What you are able to see is a very good screen, colorful. What you're able to see is good flowers, good decoration, good food, good arrangement, etc. What are the efforts behind that? So I directly go into the topic. ఉండటానికి 18 అధ్యాయాలు ఉన్న 700 కన్నా పైగా పద్యాలు ఉన్న భగవద్గీతలో అందరికీ బాగా తెలిసిన పద్యం ఒకటి ఉంటది. కర్మణ్యే బాధికాలస్తే మా ఫలేషు కదాచన మా కర్మ ఫలకేషు పోవు మాతే సంగోత్సవ కర్మణి. సింపుల్ గా చెప్పాలి అంటే ఫలితం ఆశించకు, పని చెయ్యి, ఫలితం నీ పని వల్ల వచ్చింది అనుకోవు, పని మాత్రం చెయ్యి. దిస్ ఇస్ ద సింపుల్ అండర్స్టాండింగ్ ఆఫ్ దట్. సో ఐ డోంట్ థింక్ we don't know what our efforts are going to give us in the future. ఇవాళ మనం చేసే పనికి రేపు ఫలితం ఏంటి అనేది మనం ముందే ఎప్పుడు చెప్పలేము. కానీ ఫలితంకి సంబంధం లేకుండా పని చేసుకుంటానే వెళ్ళాలి. ఐ థింక్ గడిచిన 2-3 వారాల్లో మేమందరం చేసింది అదే. ఈ కాన్ఫరెన్స్ ఎంత పెద్ద హిట్ అవుతుందో, ఎంత పెద్ద ఏమవుతుందో మాకు తెలియదు. బట్ వన్ థింగ్ దట్ వీఆర్ వెరీ ఫోకస్డ్ ఇస్ వాట్ ఆర్ ద బెస్ట్ ఎఫర్ట్ దట్ వీ కెన్ పుట్ సో దట్ the conference will go on a smooth platform without any obstructions or without any difficulties. So I would like to put forward all the efforts made by everybody. So ఎవరి పేరు మిస్ అవ్వకూడదు అని చెప్పి committee by committee I brought the book. So that's all. So ఇప్పుడు I think తల్లిదండ్రులు పిల్లలకి చేసే దాంట్లో ప్రయత్నమే ఉంటది, ఫలితం ఉండదు. అలాగే మెజారిటీ ఆఫ్ ద టైమ్స్ ఒక ఉపాధ్యాయుడు విద్యార్థి కోసం చేసే దాంట్లో ప్రయత్నమే ఉంటది, ఫలితం ఉండదు. ఈ 2 సందర్భాల్లో ఫలితం చూసి ప్రయత్నం చేసామా మోసపోతాం, సమాజమే కూలిపోతుంది. సో మేము కూడా అంతే చేస్తామని నేను గట్టిగా నమ్ముతున్నాను. ఫలితం ఏదైనా కానివ్వండి, కాన్ఫరెన్స్ ఎటైనా వెళ్ళనివ్వండి, కానీ మా ప్రయత్నంలో ఎలాంటి లోపం ఉండకూడదు అని చెప్పి మేము చాలా దృఢంగా ఒక సంకల్పంతో ముందుకెళ్ళాము. ఈ 2-3 వారాలు దృఢంగా సంకల్పం చాలా పెద్ద పదంలో కనిపించవచ్చు, బట్ ఆ 2 పదాలు మేము చేసిన పనికి తక్కువ నేను ఫీల్ అవుతున్నాను. ఫస్ట్లీ వెరీ బిగ్ అప్రిసియేషన్. అంటే ముందే చప్పట్లు ఎవరు కొట్టద్దు, అయిపోయిన తర్వాత చప్పట్లు కొట్టినప్పుడు కొడితే చాలు. అంటే నన్ను ఇంట్లో చేయదు అని చెప్తున్నాము. ఫస్ట్, as you all know, the person who is behind the entire conference is our principal, Dr. S. Vidyadhara. పెద్ద పెద్ద స్టేట్మెంట్ చెప్పాలి కానీ ఒక చిన్న ఎగ్జాంపుల్ చెప్పేస్తాము. I think 2 weeks ago or 10 days ago, some faculty members went to a conference in Hyderabad. Sorry, I cannot recollect that name. అక్కడ రిఫ్రెష్మెంట్స్ కోసం కిట్ బ్యాగ్ లో ఏదో ఒక చిన్న రిఫ్రెష్మెంట్ ఇచ్చారంట. సో దట్ అట్లీస్ట్ ఇన్ కేస్ ఇఫ్ దేర్ ఇస్ ఎనీ డిలే people can live on that. So our principal liked that idea and implemented it right here. I hope everybody received your papers in your kit bag, right? So that is how our principal will think. Wherever he goes, if he finds something that is new, he wants to implement right here, right now. And this is one way of telling you how he wants to implement on a very regular basis. And that is how, so that is how he was very, very particular about, he was very, very particular about అంటే ట్రాన్స్ఫర్మేషన్ లో మనం ఆలోచిస్తే కాన్ఫరెన్స్ ఎంత ఆలోచించాలో ఆలోచన మీకు రావాలి. So, and I think this is the first time he had a lot of confidence on all of us, because 3 days before the conference he was not even in Guntur. He went as a NAC peer team member and he was not there for the 4 days or 5 days. I think this is the first time he trusted us on so much that అంటే జనరల్ గా అనుకుంటారు కదా సార్ రాగానే మాకు తిట్టుకోవట్లేదు అంటే మేము చాలా బాగా పనిచేసామని చెప్పి. He was very, very confident with our work, and sir, thank you so much for having the confidence in all of us. Thank you for directing us. One big round of applause. Next, I am Tarvata, organizing secretary. Doctor ఆయన ఎంత ఒత్తిడికి లోనయ్యాడు అంటే గడిచిన 5 రోజుల్లో ఆయన భయపడటం చూశాను, ఆయన కంగారు పడటం చూశాను, ఆయన చిరాకు పడటం చూశాను, ఆయన భావోద్వేగానికి గురవడం చూశాను, ఆయన అయోమయానికి లోనవడం చూశాను, ఆయన అడవడం చూశాను. ఎంత ఒత్తిడికి ఒక మనిషి లోనవ్వడం పోతే ఎన్ని భావోద్వేగాలు ఎన్ని నవరసాలు కనిపిస్తాయి. So that was the effort, that was the pain, that was the amount that he underwent during this 4-5. I don't think it is 4-5 days because 20 days. That is what he has underwent in all these days. So ఇంతకన్నా ఎక్కువ చెప్పను, ఆయన ఇప్పటికే సింపుల్ గా ఉంది కరెక్ట్. అనుకోవాలి. So one very good round of applause to the organizing secretary probably of the month. And sorry sir and sorry Vikram Chaudhary sir, I'm speaking in little bit in Telugu because it's very good, it's very fast to speak in English, but to मातृभाषा में जो कीमत होती है वो ज़्यादा होती है. Next వచ్చేది obviously scientific committee. అన్ని నా దృష్టిలో అంటే జనరల్ గా ఓట్ ఆఫ్ థ్యాంక్స్ చెప్పేటప్పుడు ఆన్ బిహాఫ్ ఆఫ్ ద ఇన్స్టిట్యూట్ అంటారు. సో ఐఎమ్ నాట్ స్పీకింగ్ ఆన్ బిహాఫ్ ఆఫ్ ద ఇన్స్టిట్యూట్, దిస్ ఇస్ మై ఓట్ ఆఫ్ థ్యాంక్స్. సో నాకు పర్సనల్ గా ఫస్ట్ గుర్తొచ్చే కమిటీ ఏంటి అంటే సైంటిఫిక్ కమిటీ, బికాస్ దట్ ఈస్ ద కమిటీ దట్ హ్యాస్ టు లుక్ ఆఫ్టర్ ఆల్ ద అబ్స్ట్రాక్ట్స్ దట్ ఈస్ కమింగ్ టు దెమ్, స్ట్రక్చర్ దోస్ అబ్స్ట్రాక్ట్స్, బికాస్ వాట్ ఎవర్ ద అబ్స్ట్రాక్ట్స్ మీకు చెప్పే ఉంటారు, మీరు పంపించిన ప్రతి అబ్స్ట్రాక్ట్ కి ఒక ISBN నంబర్ అనేది ఉంటుంది. సో వాటిని స్ట్రక్చర్ చేయాలి. కొంతమంది స్ట్రక్చర్ చేసిన అబ్స్ట్రాక్ట్స్ పంపిస్తారు, కొంతమంది స్ట్రక్చర్ చేయని అబ్స్ట్రాక్ట్స్ పంపిస్తారు. కొన్నిటికి తల ఉంటే తోక ఉండదు, కొన్నిటికి తోక ఉంటే తల ఉండదు, తల తోక ఉంటే కొన్నిసార్లు ముందు తోక వస్తుంది, తల తర్వాత వస్తుంది. సో వీటన్నిటిని అరేంజ్ చేయడం అనేది చాలా చాలా పెద్ద టాస్క్. ఐ థింక్ ద సైంటిఫిక్ కమిటీ టుక్ ద హెర్క్యూలియన్ టాస్క్ ఆఫ్ లుకింగ్ ఇన్ టు ఆల్ దోస్ అబ్స్ట్రాక్ట్స్, సార్టింగ్ దెమ్ అవుట్. మళ్ళీ అబ్స్ట్రాక్ట్స్ చూస్తే సరిపోదు, వాటిని మళ్ళీ సైంటిఫిక్ కమిటీ ప్రకారం డివైడ్ చేసుకోవాలి. కాలేజీ ఏంటి, సుఫిక్స్ ఏంటి, ఇవన్నీ డివైడ్ చేసుకోవాలి. చేసుకుంటే సరిపోదు, ఏది ఓరల్ పోస్టర్ ఏది ఈ పోస్టర్ అని చూసుకోవాలి. చూసుకుంటే సరిపోదు, ఏది ఏ రూమ్ లో పెట్టాలో తెలుసుకోవాలి. చూసుకుంటే సరిపోదు, దానికి ఎడ్యుకేట్ ఎవరు అని తెలుసుకోవాలి. తెలుసుకుంటే సరిపోదు, వాళ్ళు మళ్ళీ రాయాలి. ఈ రెండు మీరు వింటున్నారంటే అర్థం వాళ్ళు చేశారు. సో ఐ థింక్ ఫర్ దట్ దేర్ ఇస్ అ వెరీ వెరీ బిగ్ రౌండ్ ఆఫ్ అప్లాస్. సో అంటే ఇందులో at the outset, the chairman for this committee is obviously Sesi sir. It is very difficult for him to manage all the things with a little discomfort to his hand, but still he took that forward. One big round of applause to Sesi sir. Next, Hanumaiah sir. According to me, the most scientific person in the scientific committee is Hanumaiah sir, and because he was the key stand of once. The please stand up once. Hanumaiah, please stand up once. Now, immediately after the conference, he has to down-regulate his anti-hypertensive dose from triple therapy to double therapy once again. Next, Haritha Pavani madam. Now, at the point of time, Haritha Pavani madam ne chuste. అందరి సినిమాలో అనుష్క ఎందుకు కూడా చెప్తాను అరుంధతి సినిమాలో ఒక సీన్ ఉంటుంది. ఆవిడని కొబ్బరికాయ పెట్టి కొడుతూ ఉంటే మేడము 11ంటికి వచ్చి చైర్ లో కంప్యూటర్ ముందు కూర్చుంటే ఎక్సెప్ట్ ఫర్ లంచ్ షీ డిడ్ నాట్ మూవ్ ఫ్రమ్ దట్ చైర్. షీ డిడ్ నాట్ మూవ్ ఫ్రమ్ దట్ చైర్. అంటే అరుంధతి సినిమాలో ఆవిడకి ఎంత దృఢ సంకల్పం ఉంటే అంత దెబ్బ తగ్గుతున్న వాళ్ళ కూర్చోగలరు. అలాగే హరిపావని మేడం కూడా అంతే, ఆవిడకి ఎంత సంకల్పం ఉంటే ఒక పక్క నుంచి వెంకటరావు సార్ వచ్చి ఎవరు కొడతారు, ఇంకో పక్క నుంచి ఇంకొకరి నుంచి ఒక్కొక్కరికే కొడతారు. బట్ షీ డిడ్ నాట్ మూవ్. I personally witnessed, I think majority of the people who are working in this conference are one very very big round of applause to Haritha. And we will stand up once. Next is Secretary sir. ఒక ఆయన చేసే పనులు ఒక 5 చేతులు కానీ 6 చేతులు కానీ ఉంటే మనందరం చేయగలము. కానీ అంటే ఇక్కడ సింపుల్ గా చెప్పాలి అంటే ఒక abstract కలెక్షన్ which is the scientific one and ఇక్కడ సౌండ్ రావట్లేదు which is the most serious one. He can deal everything with it. He can be a scientific person, he can be a computer operator, he can be, I don't know how many of you know this or not. All the banners that were designed was taken care by him only. All the decorative banners that were made by him only. That's why I said, I have changed on the Pharmaceutical Day. I am related to one very big round of applause to check on the next committee is the registration committee. We are all going to collect, but control everything. But everybody, but what's the answer? Generally, మన కాలేజీలో ఉన్న వాళ్ళందరికీ ఒకటి తెలిసే ఉంటుంది. రాము సార్కి ఒక పని చెప్తే దాన్ని మళ్ళీ తిరిగి చూసుకోవక్కర్లేదు. Once you give it the work, you need not look back at it. That is the excellence that this man carries out, and he has done the same. And one more person who fits into that category. ఇచ్చిన ఒక పని చెప్తే మళ్ళీ తిరిగి మనం చూసుకోక్కర్లేదు అన్న కేటగిరీలో ఇంకొకటి వస్తారు. అది సందీక్షలు వాళ్ళిద్దరు ఒకే కమిటీలో ఉంటే ఇంకా కమిటీ గురించి మనం ఎందుకు మాట్లాడుకోవాలా? అక్కర్లేదు కదా. So one very big round of applause to Sandeep sir and Rahul sir. Thank you, Sandeep sir. In his comment, and to add little more expertise into it, or to add little more to it, we also have Chowdavaram in the same room, same reception committee. She also did a lot of work. అంటే విజేత మేడం దగ్గర కాబట్టి ఎప్పుడైనా సరే అర్జెంట్ గా కావాలంటే ఫోన్ చేసి విజేత మేడంకే. సో ఐ థింక్ షీ ఆల్సో మేడ్ హర్ ప్రెసెన్స్ సో వాల్యుబుల్ డ్యూరింగ్ ది ఎంటైర్ ప్రాసెస్. వన్ వెరీ బిగ్ రౌండ్ ఆఫ్ విజేత మ్యామ్. I request Vijay Raman to please come inside once. And next, according to me, as I said, according to me, one of the underrated person in this institute is Mallikarjuna sir. I think he is a man with a lot of scientific expertise to himself. He is also working in the registration committee. He was the one who made all the abstracts into book, get them binded, get them to distributed on the exact day. One round of applause goes to Mallikarjuna sir also. And the next committee is the transportation committee. పెద్దగా తెలియదు, అన్ఫార్చునేట్లీ దీని గురించి పెద్దగా ఎవరో తెలియదు. ఒక చిన్న ఉదాహరణ చెప్పాలంటే, యాజ్ ప్రిన్సిపల్ రైట్లీ సెడ్, బికాస్ ఆఫ్ వెరీ అన్ఫార్చునేట్ సిచ్యువేషన్ వి వర్ డిలేడ్ లాంగ్ బ్యాక్. సో నిన్న రాత్రి 9:30 10 అయితే మనం అందరం వెళ్ళేసరికి, బట్ ట్రాన్స్పోర్టేషన్ కన్వీనర్ విశ్రామ్ గారు పొద్దున్నే 4:30 లేచి కార్ అండ్ పోలీస్ విజయవాడ ఎయిర్పోర్ట్ వెళ్ళి, దాని తర్వాత విజయవాడ రైల్వే స్టేషన్కి వెళ్ళి వీళ్ళందరినీ పిక్ చేసుకుని పైకి అలాగే ఆయన ఆ పనిలో బిజీగా ఉంటే ఇక్కడ బస్సులు అంటే బయట కాలేజ్ వాళ్ళు చాలా మంది వస్తారు. అండ్ ఇట్ బికమ్స్ అవర్ రెస్పాన్సిబిలిటీ టు ప్రొవైడ్ ద అప్రోప్రియేట్ ట్రాన్స్పోర్టేషన్. బయట కాలేజ్ వాళ్ళు ఎప్పుడు ఏ సమాధానం అడిగినా సరే వాళ్ళందరికీ సమాధానం చెప్పిన సైనులు సార్కి, అశోక్ సార్కి stand up sir. Ashok sir, if he is here. Next is the accommodation committee. So accommodation అంటే జస్ట్ వచ్చే వాళ్ళకి ఏదో ప్లేస్ చూపిస్తే చాలు అని అనుకుంటాము. బట్ accommodation is more than that. First of all, you have to make sure that you are calling everybody. Because they won't call us for accommodation. So you are calling all the 500 people who are coming here, making sure that whether they are comfortable or whether not comfortable. If they are comfortable, what place can we put there? Hostel of పెట్టాలంటే ఈజీ కాదు. లెటర్ రాయాలి, వాళ్ళ దగ్గర పర్మిషన్ తీసుకోవాలి, అప్రూవల్స్ తెచ్చుకోవాలి. ఇవన్నీ సింగిల్ హ్యాండెడ్ గా చేసిన అరుణ్ సార్, కోటయ్య సార్, and we have let's start it, Arun. So please stand up if you are here. Arun sir, Kottaya sir. Thank you once again for your services. And the most important comment. Theory వేరు, practical వేరు. Theory లో what is the important committee and the scientific committee. Practical law, what is the important committee and the hospitality committee? Hospitality committee ఎందుకు అంత ఇంపార్టెంట్ అంటే unless you provide them with the basic hospitality. అందరూ hospitality అంటే ఫుడ్ ఒకటే అనుకుంటారు. కాదు. Hospitality is right from the point they come into our institute. Are we guiding them properly? Are we making sure that they are placed in the exact positions all the time? If they are confused, what are we providing them? So that was 24/7 surveillance chesina Chowdavaram sir, Yesmani sir, Sultana ma'am, and Janki Rani ma'am. Third pair actually transportation committee lo undi, hospitality committee lo undi. Sir multiple committees lo untaru. Kakapothe sir enti ante ekkadunna sare అక్కడ చేయాల్సిన పని అక్కడ చేయాల్సిన వాళ్ళకన్నా ఎక్కువ చేస్తారు. అది ఎవరో చెప్పక్కర్లేదు, నేను ఏడుకుంటాను. ప్లీజ్ స్టాండ్ అప్ సార్. One small example, రాత్రి బాగా లేట్ అయిపోయింది, 9:30 అయింది, అండ్ ఐ థింక్ it is very sure ఎప్పుడైనా లేట్ అయితే our management, our principal, they are very particular. లేట్ అయితే పిల్లలకి ఏదో పెట్టి పంపించాలి, డిన్నర్ లేకపోతే ముందే ముగించాలి అన్న ప్లానింగ్ ఎప్పుడు ఉంటుంది. కానీ అన్ఫార్చునేట్ గా జరగలేదు. సో నిన్న అర్ధరాత్రి 9:30కి ఫ్రూట్ షాప్స్ ఉంటాయేమో చూడమన్నారు. ఫ్రూట్స్ అమ్ముకునే వాళ్ళు ఉంటారు కదా ఆ టైంలో, ఎవరు ఉంటారు ఇంకా. బట్ సార్ అక్కడ లేదు, మొత్తానికి ఎక్కడికి వెళ్ళాలో తెలియదు, ఏం చేశారో తెలియదు, మొత్తం బిస్కెట్లు అన్ని పట్టుకొచ్చేసారు. And the next one, state committee and Uttarasevi Subhash and next అంటే సుబ్బారావు సార్ ఎక్కడ ఉంటే మనం అందరం ఇక్కడ పనిచేయాలంటే సుబ్బారావు సార్ చూసైనా పనిచేయాలనిపిస్తుంది. సుబ్బారావు సార్ కోసమైనా పనిచేయాలనిపిస్తుంది. సుబ్బారావు సార్ వల్ల అయినా సరే మనం ఎక్కువ మంది పనిచేయాలనిపిస్తుంది. కానీ సుబ్బారావు సార్ అంటే మాత్రం పని జరుగుతుంది. In that way, he manipulated everybody. Student level, staff level, people level, everyone అందరినీ ఆయన manipulate చేశారు. So for me, Subharu sir is a very I don't know how many of you are aware of this concept called as Dabbawalas of Mumbai. Subharu sir అయిపోయింది ఇంకా నెక్స్ట్ ముష్కిల్ Dabbawalas of Mumbai ఎవరికైనా తెలుసా? వాళ్ళు ఏం చేస్తారంటే ఇంటింటికి వెళ్ళి క్యాషెస్ కలెక్ట్ చేసుకుని ఎవరైతే వర్క్ చేస్తున్నారో ఆఫీసెస్ లో వాళ్ళు డిస్ట్రిబ్యూట్ చేస్తారు. దానికి ఒక గెజెస్ బుక్ ఆఫ్ వరల్డ్ రికార్డ్స్ కూడా ఉంది. ఏంటంటే వాళ్ళ దగ్గర అడ్రస్ లే ఉంటారు. ఒక 10 మంది 20 క్యారేజెస్ సైకిల్ లో పెట్టుకుంటారు. అవన్నీ ఒక పడవలోకి ఎక్కిస్తారు. పడవలో వేల క్యారేజెస్ ఉంటాయి. అవన్నీ మిక్స్ అవ్వకుండా, మళ్ళీ వాటి పేర్లు ఉండవు, అవన్నీ మిక్స్ అవ్వకుండా ఎవరి దగ్గరికి వెళ్ళాలో వాళ్ళ దగ్గరికి వెళ్తాయి. ఒక మినీ అటెంప్ట్, అదే అంత ఎఫెక్ట్ కాకపోయినా ఒక మినీ అటెంప్ట్ సింధు వైష్ణవి మ్యామ్ అండ్ రికార్డ్ మ్యామ్ చేశారు. చూసే ఉంటారు, ఒక్కొక్కరికి దగ్గర 3 నుంచి 4 హెల్త్ స్టేషన్స్ జరిగాయి. అంటే ఒక్కొక్కరికి 3 నుంచి 4 మూమెంట్ వెళ్ళాయి, ఒక్కొక్కరికి 3 నుంచి 4 స్టాల్స్ వెళ్ళాయి. కానీ ఎవరికి ఇంకొకరితో కలవలేదు. ఎవరి మూమెంట్ వాళ్ళకి వెళ్ళింది, ఎవరి స్టాక్ వాళ్ళకి వెళ్ళింది. మోస్ట్ ఇంపార్టెంట్ ఎవరి పనులు వాళ్ళకి వెళ్ళిపోయింది. అది సార్ట్ అవుట్ చేయడం ఎంత కష్టం అనేది కంప్యూటర్ లెవెల్ రూమ్ లో ఉన్న వాళ్ళకి మాత్రమే తెలుస్తుంది. అండ్ అఫ్ కోర్స్ అప్పటికప్పుడు సడన్ గా కొంతమంది గెస్ట్ వస్తారు, మన దగ్గర ఉన్న రిసోర్సెస్ కొన్ని వాళ్ళకి ఇవ్వాల్సి వస్తుంది, దాన్ని మళ్ళీ ఫుల్ బ్యాక్ చేసుకోవాల్సి వస్తుంది. ఐ థింక్ దిస్ పీపుల్ డిడ్ ఎ వండర్ఫుల్ జాబ్. సింధు వైష్ణవి మ్యామ్ అండ్ రిహానా మ్యామ్, I hope Rimana ma'am is here. One very big round of applause to all of them. And కమిటీలో ఉన్నవాళ్ళు మాత్రమే అని కాదు, there are of course we all know that there are examinations happening to fourth form students. So ఉన్నవాళ్ళందరూ కమిటీలో ఉంటే అయ్యారు చూసుకోవాలి. So there are other people, even though they are not directly involved in this conference, they made sure that when these people are not available, they made sure that everything else is happened properly in the college. So one very big round of applause to all the people whom I did not mention. One very big round of applause to all the people who did not mention. You made a very important role. Andhra Pradesh యొక్క పిల్లలు పడతారు కాబట్టి నేను పనిచేసుకోగలిగామని నేను నమ్ముతాను. And most importantly students చెప్పిన ప్రతి పని చేశారు. ఎంత బాగా చేశారంటే చాలా సార్లు నేనే అనుకున్నాను వీడు క్లాస్ లో చెప్పినప్పుడు ఎందుకు చేయలేదు అనిపించింది. Of course అదే రోజు చేస్తాను. బట్ దే డిడ్ వండర్ఫుల్ జాబ్. ఏం చెప్తే అది చేశారు. ఒక చిన్న ఎగ్జాంపుల్ చెప్తాను, పేరు మెన్షన్ చేస్తే ఒకళ్ళే చేశారని ఇంటెన్షన్ వస్తుంది. ఐ థింక్ మీ అండ్ వెంకట్రావు సార్ అంటే ఇన్స్ట్రుమెంటేషన్ ల్యాబ్ దగ్గర నుంచి ఆఫీస్ కార్డినేటర్ నడుస్తున్నప్పుడు ఒక అబ్బాయి వచ్చి ఇద్దరికి వాటర్ ఆఫర్ చేశాడు. నేను అడగలేదు, సార్ అడగలేదు. నేను తాగిన తర్వాత ఎందుకమ్మా ఇచ్చావు అంటే, సార్ పని చేస్తున్నారు కదా సార్ కొంచెం తాగుతారని చెప్పి ఇచ్చాము అన్నాడు. ఐ థింక్ దట్ ఈస్ డెడికేషన్ all of the students who are in different volunteers. I think now all the staff here మనం సపోర్ట్ కొట్టాలి వాళ్ళకి. వన్ వెరీ బిగ్ రౌండ్ ఆఫ్ అప్లాస్ టు ఆల్ ఆఫ్ దెం. అంటే ఎంత హడావుడితో మాకు తెలుసు, ఎందుకంటే మనం ఒక ప్లానింగ్ అనుకుంటాం, సడన్ గా స్పీకర్ రాదని తెలుస్తుంది, సడన్ గా స్పీకర్ మారిపోయాడని తెలుస్తుంది, అప్పుడు మాకు చాలా కంగారు ఉంటుంది, ఏం చేయాలో తెలియదు. ఎదురుగా మీరు కనిపిస్తారు, గట్టిగా అంటామో ప్రేమతో అంటామో ఏమంటామో తెలియదు, నాకు మాట్లాడేస్తాం. బట్ మీరు ఆ మాటలన్నీ పట్టించుకోకుండా ఐ థింక్ యూ డిడ్ అ వెరీ వండర్ఫుల్ జాబ్. ఇఫ్ పాసిబుల్ రోజు క్లాస్ లో పాటలు చేసుకుంటున్నాను, అది కష్టం అని తెలుసుకోవచ్చు, ప్రయత్నం చేయండి. అలాగే వాళ్ళు అలవాటు అయింది కదా తిన్నాను వాళ్ళు, రేపటి నుంచి ప్రయత్నం చేయండి. వన్ వెరీ థ్యాంక్ యూ ఆల్ ద స్టూడెంట్స్.
వాటర్ ఇస్తారే
ఇప్పుడు వాటర్ థ్యాంక్ యూ ఆల్ టు ద స్టూడెంట్ మెంబర్స్. అండ్ నెక్స్ట్ కమింగ్ టు ద నాన్ టీచింగ్ స్టాఫ్. నాన్ టీచింగ్ స్టాఫ్ అనగానే చాలా పేర్లు గుర్తొస్తాయి. ఫస్ట్ చెప్పుకోవాల్సిన పేరు సరిత మేడమే. ఎందుకు అంటే ప్రోగ్రామ్ షీట్ 10 సార్లు మారుతుంది, 10 సార్లు మార్చేది ఆవిడే. ప్రోగ్రామ్ షీట్ అనేది రోజు ఒకటి ఉంటది, చేసేది ఆవిడే. దాని తర్వాత ఎన్నో స్పీచెస్ టైప్ చేయాల్సి వస్తుంది, ఎన్నో ట్రాన్సాక్షన్స్ చూడాల్సి వస్తుంది. ఐ థింక్ వన్ వెరీ బిగ్ రౌండ్ ఆఫ్ అప్లాస్ గోస్ టు సరిత మ్యామ్. ఇట్ ఈస్ హియర్. One very big round of applause to Chandra. And we all know that conducting a conference is a very costly affair. I think the screen that you are seeing costs more than 1 lakh. So there are a lot of transactions that will be happening whenever you are doing it. Food and decoration and transportation and chala costs untayi. Avanni maintain cheyadam anత easy kaadu. I think our L. Venugopal sir, he also deserves a very big respect for making all these transactions possible. Okay. And also all the lab assistants and also all the lab assistants, both male and female lab assistants, they are very helpful. Practical కోసం practical జరుగుతున్నప్పుడు మేము ఎప్పుడైనా ప్రిన్సిపల్ కి ఏమన్నా మీటింగ్ పిలిచారో వెళ్ళిపోతే వాళ్ళ మీద వదిలేసి వెళ్ళిపోతాము. They made sure that the practicals went on. They made sure that the lab was taken care of. And sudden గా ఏమన్నా కావాలన్నా సరే వాళ్ళకి పంపించి వాళ్ళని హెల్ప్ చేయమని అడుగుతాము. వాళ్ళు చెప్పిన ప్రతిదీ చేశారు. One very big round of applause to all students staff. And somebody who we should not forget are the second grade and third grade workers, because they are very important in keeping this premises very clean, making this premises very amiable. So guests ఎవరైనా వచ్చినప్పుడు ఎంత క్లీన్ గా ఉంచాలి అనేది వాళ్ళ చేతుల్లోనే ఉంది. చెప్పిన ప్రతిసారి they kept on doing it. One very big round of applause to all the workers. And with this we came to the last 2 minutes of vote of thanks. Last 2 minutes of vote of thanks. Next question. ఇందాక నేను చెప్పినట్టే ప్రయత్నం ఎందుకు చేస్తున్నాం ఈ ప్రయత్నం. సో వాట్ ఈస్ ద ఇంటెన్షన్ బిహైండ్ డూయింగ్ అ వెరీ బిగ్ కాన్ఫరెన్స్? వన్ సింపుల్ కట్ సొల్యూషన్ ఆర్ వన్ సింపుల్ కట్ ఆన్సర్ టు ఎన్హాన్స్ ద నాలెడ్జ్ ఆఫ్ ద స్టూడెంట్. యాజ్ సింపుల్ యాజ్ దట్. ఇంకా అంతకన్నా పెద్దగా గొప్ప గొప్ప ఆలోచనలు అయితే ఎవరికి ఉంటాం. నెక్స్ట్ థింగ్ ఇస్ ద ఎఫర్ట్ దట్ వీఆర్ పుట్టింగ్ ఇన్. ద ఎఫర్ట్ దట్ వీఆర్ పుట్టింగ్ ఇన్. నిన్న మీకు 9:30 అయింది, మాకు 5 రోజుల నుంచి 9 అవుతుంది, రైట్? బట్ అవి ఎప్పుడు చెప్పుకోలేదు ఇంత లేట్గా వెళ్తున్నాం, ఇంత పని చేస్తున్నాం అనుకుని. బికాస్ యాజ్ ఐ సెడ్, వీఆర్ నాట్ వరీడ్ అబౌట్ ద అవుట్కమ్. వీఆర్ ఓన్లీ వరీడ్ అబౌట్ వాట్ ఎఫర్ట్స్ కెన్ బి పుట్ ఫార్వర్డ్ ఫర్ దిస్ ఈవెంట్ టు హ్యాపన్ ఆన్ అ వెరీ బిగ్ స్కేల్. దట్ ఈస్ వాట్ వీ వర్ డూయింగ్. అండ్ ఫలితాన్ని ఆర్పించకూడదు, కానీ చిన్న కోరిక అయితే ఉండొచ్చు కదా, ఇలా అయితే బాగుంటుంది కదా, ఇలా చేస్తే బాగుంటుంది కదా అని చెప్పి. అండ్ everybody on the stage will agree with me. Conferences are very, very important for the upliftment of any education institution. Forget pharmacy, forget engineering, forget arts and sciences. These conferences that are very, very important, because whatever the insight that we have, or whatever the insight that we have when we read our books or when we do our practicals, conferences gives us a bit beyond that. మన పుస్తకాల్లో చదవండి లేదేదో తెలుసుకోవాలి అన్న ప్రయత్నం కాన్ఫరెన్స్ చేస్తుంది. అందుకనే కాన్ఫరెన్సెస్ కూడా కండక్ట్ చేస్తాము. సో ఐ థింక్ ఇది అందరికీ తెలిసే ఉంటుంది. త్రివిక్రమ్ అనే ఆయన సిరివెన్నెల సీతారామ శాస్త్రి గొప్పతనం గురించి చెప్తూ ఒక చిన్న స్పీచ్ ఉంటుంది. ఇప్పుడు అదంతా చెప్పిన ఒక చిన్న మాట చెప్తాను. ఆయన ఎందుకు అంత గొప్ప కవి అయ్యారు అంటే, ఒక హీరోకి కలర్ బట్టలు వేసుకుని డిస్కో డాన్స్ చేయాలని ఉంటుంది. ఒక డైరెక్టర్ కి ఫారెన్ పోయి అక్కడ షూటింగ్ చేయాలని ఉంటుంది. ఒక ప్రొడ్యూసర్ ఒక ఐటమ్ సాంగ్ పాడితే డబ్బులు వస్తాయి కదా అని ఉంటుంది. సో అక్కడ అదంతా ఇరుకు ఉంటుంది. అంత ఇరుకులో కూడా ఆయన మంచి సాహిత్యాన్ని రాయగలిగారు, అందుకని ఆయన అంత గొప్ప వాళ్ళు అయ్యారు అంటారు. సరే, ఇప్పుడు ఈ పోలిక ఎందుకు చెప్తున్నాము అంటే, as Vasudeva said, my book is not here, but he said there is always a scope for betterment. There is always a scope of betterment. So ఇప్పుడు ఏంటి అంటే, పరిశోధన దృక్పథం కన్నా ఒక ధృవీకరణ పత్రం ఉంటే చాలు. ధృవీకరణ పత్రం అంటే సర్టిఫికెట్. ధృవీకరణ ఒక పరిశోధన తెలుసుకోవడం కన్నా ఒక ధృవీకరణ పత్రం ఉంటే చాలు. లేదంటే శాస్త్రీయ ఉపన్యాసం కన్నా సినిమా పాటలు ఉంటే చాలు. లేదంటే సాంకేతిక శాస్త్రీయ మేధోమదనం జరగాల్సిన ఒక చోట చాలా అల్పమైన విధులు వినోదాల గురించి జరుగుతున్నప్పుడు ఒక చిన్న బాధ కలుగుతుంది. అంటే ఒక కాన్ఫరెన్స్ నుంచి బయటికి వెళ్ళిపోయిన తర్వాత, ఈ కాన్ఫరెన్స్ లో జరిగిందంటూ ఆయన అంటారు కదా, ఒక ఎగ్జైట్మెంట్ తో పడుకోవాలి ఆ రోజు రాత్రి అని, 2 చేతులు జేబులో పెట్టుకుని నడుచుకుంటూ వెళ్ళిపోవాలి అని చెప్పి. సో అలాంటిది ఏ ఒక్క చిన్న కోసాల జరిగినా సరే మేము సక్సెస్ అయినట్టే. As the team chips, as team Operant Pharmacy Federation, we are very, very successful if that thing happens. But ఇంత ఇరుకులో చెప్పాను కదా, సో ఇంత ఇరుకులో కూడా మేము ఎంత ప్రయత్నం చేస్తున్నాము అంటే, ఎక్కడో ఒక చోట ఎవరో ఒకరికైనా సరే ఆ రోజు ఒక ఎక్సైట్మెంట్ తో పడుకుంటారేమో అని చెప్పి. సో ఇది ఫ్యూచర్ లో ఇంకా ఇంకా జరగాలి. ఒక విద్యార్థికి వాడికి ఏం కావాలి అన్నదాన్ని పక్కన పెట్టేసి, వాడికి ఏం అవసరం అన్నది ఇవ్వాలి అనే ఒక ప్రయత్నం నిరంతరం జరగాలి. అది మన నినాదం కావక్కర్లేదు, కనీసం మనకు ఆవేదన ఉండాలి. వాడు ఏం కావాలో వాడు సినిమా పాటలు తెలుస్తాం, బేబీ కొట్టడు తెలియలేం కదా. సో వాడికి ఏం కావాలి అనేది పక్కన పెట్టేసి, వాడికి ఏం అవసరము అన్న ప్రయత్నం మనందరం నిరంతరం చేయాలి. నేను చెప్పినట్టు అది మన నినాదమే అవ్వక్కర్లేదు, కానీ మనకు ఆవేదన అయితే ఉండాలి. ఇలాంటిది ఫ్యూచర్ లో చాలా చాలా జరగాలని చెప్పి. ఇట్ ఈస్ ఆల్వేస్ a pessimistic way to complain. It is always a pessimistic way to complain. The optimistic way is hope. I am very hopeful that Chebrolu from my Institute of Pharmaceutical Sciences, with this wonderful team, will come with multiple conferences in the future, so that we can give you better outcomes at every point of time. Thank you once again. Please stand for the flag.
Sorry to disturb you. వీళ్ళందరూ చెప్పాడు. ఇప్పుడు 2008 నుంచి రాఘవ కళ్యాణ్ నా పక్కనే ఉంది. 2008లో వాడు ఇక్కడ జాయిన్ అయ్యాడు. బీఫామ్ అయింది. తర్వాత ఫార్మ్ డి ఏఎస్ఎస్ మైసూర్లో చదివాడు. మళ్ళీ ఇక్కడికి వచ్చి ఇక్కడ ఫ్యాకల్టీగా జాయిన్ అయ్యాడు. మధ్యలో 3 ఏళ్ళు గ్యాప్ తప్పితే 2009 నుంచి నాకు ఎనీ ఫంక్షన్ ఏది జరగాలన్నా మోస్ట్ కాన్ఫిడెంట్ అండ్ ట్రస్ట్ వర్తీ పర్సన్ నాకు ఎవరైనా అంటే వాడు ఉంటేనే ఇక్కడ అలంకారం. అలాగే చక్రవర్తి వాడు ఇక్కడ స్టూడెంటే. సో ఇక్కడ ఒకరి దాన్ని స్పెసిఫిక్ గా చెప్పడం కాదు, వాడి గురించి వాడు చెప్పుకోలేదు. ఇక్కడ వాడు ఉన్నాడు కాబట్టి ది ఎంటైర్ సైంటిఫిక్ deliberations are went on very smoothly. Every 5 minutes change అవుతుంది. గెస్ట్లు ఎవరు వస్తారో తెలియదు, గెస్ట్లు కొత్త వాళ్ళు వస్తారో కొంతమంది డ్రాప్ అవుతారో తెలియదు. ఎవ్రీ 5 minutes కి పిలిచి వీళ్ళని తీసేయి, వీళ్ళని యాడ్ చేయి, పెట్టు, ఈ ప్రోగ్రామ్ వద్దు, ఇది తర్వాత మార్చు అని ఎప్పటికప్పుడు షడ్తా ఉండేవాడు. ఎవ్రీథింగ్ he could able to organize very well and in a systematic way. That is what our entire chiefs team, not only Ramakalyan, Chebrolu, Subbarao, many others, every staff member and every student, this time they made it real and you all rocked it. Thank you.
All the student volunteers, please stay back for group photo. All the student volunteers of various committees, please stay back for group photo.
Registration is reception and last certificate. Or certificates give back.
ಹೇಗೆ ನನಗೆ
తర్వాత ఇది